Welcome, everyone. My name is Dan Smale. I'm a professor in the history department and a member of the steering committee of the Science of the Human Past. And it's my really distinct honor to have a chance to welcome all of you, including our distinguished visitors and guests, to the inaugural lectures this afternoon. What we're going to be doing is having relatively short talks, about 20 minutes with 10 minutes of questions and answers afterward. So I'll be moderating those discussions. I'll have to leave myself at about 3:45, and my colleague Joyce Chaplin will step in to chair the few remaining panels. I'd like to invite those of you in the back to come in and sit in these chairs. They did say reserved, but they're now reserved for you. So please come in and populate these chairs. We want to make sure that everyone is comfortable and has a chance to really engage with the wonderful things that we'll be doing this afternoon in all these talks. So now it's just another real privilege to welcome and introduce my friend and colleague, Mike McCormick. When I first met Mike, it was probably about 14 years ago, and I remember in one of our first sustained conversations, we were crossing Harvard Yard, and Mike turned to me and said, what would you do if you had a good deal of financial resources and could do anything you wanted to in the medieval past? I'm another medieval historian. And I didn't realize at the time that this is actually the question that he himself was facing. So I remember thinking, I don't know if you remember this, Mike, I remember saying, because I was reading about the conversion of Jews in Iberia in the later Middle Ages, and I remember saying, but I'd be really fascinated to find out with the new techniques that we have, what percentage of the population of the Iberian Peninsula actually has a background. And so it was one of those, I had no idea that this is the direction that Mike was going to be heading at this time. So I've been sitting as an admiring spectator and often enough participant as this amazing project has been developing here at Harvard, and now in collaboration with our colleagues at the Max Planck Institute. Mike McCormick was born on the banks of the Erie Canal and received his PhD from the Université Catholique de Louvain in 1979. And he served on the faculty of the Department of History at Johns Hopkins University from 1979 to 1991. And was the research associate at Dumbarton Oaks from 1979 to 1987. He came to Harvard in 1991, and he's currently the Francis Golett Professor of Medieval History, and of course, as you know, chairs the initiatives we have in the science of the human past. He is an extraordinarily distinguished medieval historian in addition to his many other accolades. He has written numerous monographs and articles. His great book, The Origins of the European Economy, Communications, Commerce, and Commerce, AD 300 to 900, came out from Cambridge University Press in 2001. And won the Haskins Medal of 2005 from the Medieval Academy of America. It has been described as the Henri Pirenne of the 21st century. It's also won the Ronke Prize of the Economic History Association for the best books in 2001 and 2002. He actually does have time to continue to publish new books. His Charlemagne's Survey of the Holy Land, Wealth, Personnel, and Buildings of the Mediterranean Church between Antiquity and the Middle Age just came out in 2011. And how he manages to do all this in the midst of everything else is anyone's guess. So let me now just welcome Mike to the podium to give his presentation.
Thank you, Dan. Thank you, Dan. Is this working now? We got it. Am I doing what I'm supposed to do in terms of sound? Can you hear me? On. Okay. Here we go. So I thought I would begin by, since we're historians, some of us are historians, by giving you a little history of how we came to be in this extraordinary moment. I think it's fair to say that we are all present at the birth of a new discipline, certainly the institutional form of a new discipline that's been percolating up from different ventures going on around the world. And in my case, with the anecdote that that Dan just referred to, the immediate impetus was this extraordinary bolt from the blue in 2002 when the Mellon Foundation dropped, totally unexpectedly, unapplied for, a massive grant on my head, allowing me to do whatever I want in research. And my children thought we should head right off to Toys R Us and buy the store. I said we couldn't spend it on toys. And my wife said, but Mike, now you can do all the crazy ideas you have. And I got advice from some very wise people on how to use such incredible found money to try things that were impossible. It didn't matter if I was going to fail. I didn't have to answer for the money. And so I tried to do many, many different things, figuring that if I tried 15 things and 12 of them failed, 3 would work. And if they were crazy enough, it would be really worthwhile. So that's where this really got started. And I brought together people who might otherwise not meet. We had a meeting, for example, in 2005 of economists, a young assistant professor in genetics by the name of David Reich. Nick Patterson was there. And we wondered whether it was possible to reconstruct economic history from ancient genetic evidence. Right from the beginning, we wanted to involve our brilliant undergraduates and graduate students in the operation. And those of you who know the field a little bit will be pleased to see that our graduate student scribes there. One of them works for Johannes in Jena right now, and the other is vice president and professor of classical studies at the University of Oklahoma. So, and just published a big book on environment, disease, and the fall of the Roman Empire. The next big step came in 2008. When the Broad Institute at MIT Harvard generously sponsored an initial meeting to explore this whole crazy idea of using the natural sciences to discover the past in a systematic way. It was a really cool meeting. We had a tremendous amount of fun, and we made some progress on some important issues. The next step came in 2013 when a group of Max Planck Institute directors in Germany found out about my work, and they knew this brilliant young geneticist by the name of Krause. They said, well, let's bring these guys together and see what happens. Some of you know what happened, the most famous train ride in history. Johannes came to pick me up at Frankfurt Airport, and we missed the stop. I noticed Phil. I said, Phil, isn't that past where we're going, Johannes? Oh, yeah. We got out, we got another train, we got back, we kept talking, we missed the stop again. We finally did get to Tübingen, where Johannes' lab included the lab of Misha, who discovered das Nuclein, DNA, in the 1860s. So that was pretty cool. And that friendship has continued to grow and prosper. Oops, and we're stuck. There we go. In 2013, we launched the Initiative for the Science of the Human Past with a conference at the Radcliffe Institute, featuring some pretty cool speakers, and a very, very good time. And it has now taken this new form, there we go, of this wonderful new institution, this institutional collaboration between the Max Planck Society of Germany, the most distinguished and accomplished intellectual enterprise in the Federal Republic of Germany, and Harvard University, where we're working together to try and bring together our two institutions to create a network of students and scholars and scientists who are talking in ways and with people that they never would have spoken with otherwise. We had a town meeting here in this very room in February to start to do the brainstorming within Harvard so that we could find out how we could be useful to people around this magnificent institution and plug into the Max Planck Society. And Johannes' brand new institute for the Science of Human History, the Max Planck Institute for the Science of Human History. So our idea was to build a virtual transatlantic bridge through Skype, through the internet, of getting across the Atlantic and trying to bring our minds together. But also to do so in human terms, because the internet's great, but if you actually know each other and have spent time together and drunk beers together, you're more likely to look each other up on the internet and be talking about routine things. So we've worked hard to build, and we are building a virtual transatlantic bridge, and so you see the graduate students from Jena are here with us for the second time this year as part of our group. The real challenge may be building another bridge across the Charles River. We're real proud of our successes in that area as well. It's a shorter bridge, but in some ways a longer bridge to the medical school and our colleagues over there. The graduate students are one of the most beautiful parts of this new enterprise. We aim to train a new generation of new minds, and we're very, very pleased to salute the first two graduate students in the program, Aurora Alshaus and Megan Michael, who are working in the human evolutionary biology and in anthropology archaeology, and Aideen Harney, who has the first dissertation research grant from the new institute. And this is just extremely cool. finding funding for graduate students is the most difficult task for any fundraiser in any major American university. And that Max Planck and Harvard have been able to come together on this with the wonderful support of our various supporters is really a major development. In 2017, we took our graduate students to Jena and worked together for a week there at the wonderful institute there, went into their labs and had some fantastic discussions about the two main research focuses of our project. The umbrella institution at Harvard for this new departure is the Initiative for the Science of the Human Past that you've heard about, that got started in 2011. And it is a network of colleagues from across Harvard University, from the Divinity School, from the Faculty of Arts and Sciences, from the medical school, from the School of Public Health. And we aim to foment and encourage breaking down the silos and bringing together people who would never meet, professors, students, researchers, from across the university and outside the university. We have some very important collaborations going with MIT at present. There are many aspects to what the Science of the Human Past does that can serve our new Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean, pronounced by some of us as MOM, others as MHAM, and still others as Mayhem. The Digital Atlas is a wonderful tool that's used by scientists, scholars around the world. It's created by Harvard undergraduates. Some of the undergraduates in this room have contributed to making it. It's free, it's available, and we use it not only as a place to visualize the past, but as a data dump. You can download all the ancient shipwrecks you want here, the whole Roman road network. Very cool thing. We have archaeological operations. We've been active in Spain using advanced techniques that allow us to detect structures under the soil without excavating. In the case of Braccopolis, we've been busy for the last 3 years in 118-degree temperature. We have a wonderful project in the isotopic Silk Road. Silk is the first really globalized commodity, moving from China across the globe into the Roman Empire, into ancient Greece. My colleague Noreen Touros, who would be with us but has a little illness today, has invented this amazing new method that allows her to determine using isotopes where on the surface of the globe a caterpillar has munched on his or her mulberry leaves. Therefore, you can take any piece of archaeological silk and figure out where it came from. For reconstructing, there's a lot of archaeological silk that survives, so for reconstructing ancient trade networks from China to Western Eurasia, this is amazing. And finally, we have the historical ice core project that we got from Switzerland in 2013. We retrieved an ice core, and we're in the process of using the ancient atmosphere trapped in that to reconstruct the economic and environmental history of Western Europe. So far from 600 AD forward, we think it goes way far back. So these are projects that are under the Science of the Human Past that can serve the new institution that we've created. This institution grows out of collaborations that had already been underway. The 2010 publication of the Neanderthal genome brought together Johannes Krause and David Reich and many of our friends. So we've been working together, the big publication identifying the genetic marker of the movement of the Indo-European speakers into Europe. Again, if you look through the list, you'll see many familiar names from the MHAM collaboration there. Johannes Krause's work has been absolutely path-breaking in recovering the ancient pandemics that devastated the economy, transformed the world of the people that were living then. And Johannes particularly has developed the genomic approach, reconstructing the entire genome of the ancient pathogens, which opens wonderful windows into questions of evolution and the history of the disease, the spread of the disease and its differential impact in different areas. Most recently, we've had already a proto-MHAM collaboration, publishing a robust, new, complete genome with deep coverage, geneticists will know what I mean, of a young man and a young woman who died of bubonic plague in Bavaria in the time of the Emperor Justinian in the 6th century. There's no written record that bubonic plague reached Bavaria, but we now know that it did. So these are the kind of things that we've been doing that have led up to this and taking us to our research focus, our dual research focus of mobility, migration, and ancient pathogens in the Mediterranean. Yosif Lazaridis is going to take us to the latest results on the genetic aspects of the peopling of the Mediterranean when he talks to us this afternoon about Minoans and Mycenaeans and the ancient DNA evidence. Mobility is not just people, it's microbes. Microbes usually don't travel under their own power, so they're part of the bigger picture of human mobility, of movement, of exchange. And ancient pathogens have much to teach us about the past. I used to ask, did plague end the ancient economy? Now, with the revolutionary results that show that bubonic plague, contrary to what we believed, actually is much older than we thought, one has to ask, how many times did plague end ancient economies? Johannes Krause is going to share with us his latest thinking on the plagues before the great Justinianic pandemic, that one that we documented from Bavaria, that brought down significant parts of the Roman economy in the 6th century. There are challenges to this extraordinary adventure we're on, and the first is that of the two cultures, humanities versus the sciences. They are two cultures today. They speak different languages. They write in different styles. Scientists think using the passive voice is really good. Humanists say, that's terrible style. Huh? We have very different ways of evaluating probability. Humanists like to use finely shaded nuance of wording, whereas scientists want numbers to explain. These are different elements of culture that we must learn to exchange and adopt to each other and learn each other's way. Susan Alcock is going to help us understand a little bit about the outcome of this kind of new and rather daring collaboration. There are challenges of scale. For the first time in the field I work in, we're dealing with data of such enormous scale that you need computers to be able to manipulate it. You need advanced algorithms to be able to see patterns. So data, big data, is what's coming out of the genomic research, out of our ICE core research. David Reich is going to share with us his experience as ancient genetics moves into an industrial scale of investigation. And then finally, the analysis. I mean, this is the whole point. It's great to have data, but data without analysis doesn't take us far. And here's another kind of paradoxical challenge. So we have just witnessed the equivalent of, I would like to say, Newtonian physics, or Einstein's theory of relativity. We now see the continents. We see that, by God, we all come from Africa. And now we're starting to see facts of smaller, of more refined, of finer details. And at first there may be some kind of disappointment. Oh my God, where else could we have come from beyond Africa? But we shouldn't be disappointed. We have to take that challenge and realize, okay, we know that there are 7 continents. Now we're going to find out about the mountain ranges and the rivers, and that's what's really going to transform our deep understanding of the human past, where the work of archaeologists and historians have already done so much fabulous work. There are challenges of resources. Not just intellectual resources, but resource as in equipment. So this little device here is described as really a bargain. It's just under $1 million. George Church says the price is plummeting. This is fantastic. Well, you know, humanists in the room, you know, you say, here, you need this $1 million for your typewriter. This is science, and science is really expensive. We've got to figure out how to assemble the resources in order to be able to do this science. It can't be done on the cheap. It can't be done any other way. So there are challenges that are quite real. But there are also opportunities. What better way to overcome the clash of the two cultures than by bringing them together, by breeding a new kind of researcher who combines the humanities with the sciences, who has all the strengths of the humanities as well as the sciences. And I was trained as a philologist, Greek philology, and that was my game. And nothing we are going to do in the discovery of new genetic, scientific evidence about the historical past, or even the prehistorical past in many respects, is going to replace, make obsolete, sovereign philological command of the ancient languages in which the earliest texts are written. It is a fact that I do not understand, that bubon, the word that comes to be bubonic plague, is a non-Indo-European word that got into Greek. That's really interesting. Ingwen, the equivalent in Latin, is apparently a non-Indo-European word. Whoa, that's really food for thought. And besides that, now science and philology must go together. There's been a stalemated debate because the molecular clock, calculating how old an organism is by the rate of evolution, microevolution within the organism, led us to believe that the organism of bubonic plague was about 1,400 years old, 1,500 years old, so it must have emerged at the time of the Justinian pandemic. We all believe that. Now, there are some Greek texts, medical texts, that seem to talk about something that sounds like bubonic plague before that. But that's impossible until a paper appeared which discovered in ancient skeletons the presence of Yersinia pestis, bubonic plague, 5,000 years ago. With that knowledge in hand, one of our graduate students went into the Thesaurus Linguae Latinae and assembled exhaustive comparanda, Greek, all passages in Greek which are in any way related to these passages, and zeroed in on the semantics of every comma, every accent in the Greek text, and has proven to my satisfaction, and I think ultimately to the world, that bubonic plague is indeed documented in texts already from around 100 AD and maybe earlier in certain areas of the Mediterranean. This will be quite important as we go forward in this field. So the humanities and the sciences actually must work together and are being obliged to work together because the sciences are destabilizing what we thought we knew from the archaeological and written evidence of the past. As far as scale is concerned, I think it's pretty good to discover mountain ranges, giant rivers, Grand Canyons. That's where we're going now. The key thing will be to bring together different disciplines. We all come from Africa. We know this. This is established genetically. But why? Why did we leave Africa? A paper was published just recently that argues that the five great waves of human migration, oops, and they faded right out here. There are five very faint bars there where the arrows point. Correspond to major climate upheavals. So we have a possible cause for the first through fifth human migration pulses out of Africa. I mentioned the amazing discovery that that links the movement of a genetic pattern from the borders of Asia and Europe into Western Eurasia. That is to say, that's been linked with the spread of the Indo-European languages. We all are speaking, I'm speaking English, which is an Indo-European language. French, German, Greek, Iranian, they're all Indo-European languages. Another study has revealed to our astonishment, we'll see what our geneticists think of it, that the genetic signal shows that the migrants were almost exclusively male. That's a completely different historical and archaeological phenomenon, human phenomenon, than the migration of the Neolithic expansion that brought agriculture to Europe a few millennia before, in which it appears it was a balanced group of men and women who moved into Europe and brought the new technology of farming. If this is correct, this means we have to go back to the archaeological record and start to think about what it means. What are we seeing? How do we see gender differences in the archaeological record that help us understand what's going on here? This, without genetics, we couldn't dare to pose these questions. By way of conclusion, what's going to happen, what is happening, what we're attempting to achieve in MHAM is to bridge the gap, to bring together the sciences and the humanities, to make the most robust kinds of observations, because they are observations that occur and are founded in different areas of reality, using totally independent methods and technologies, and that converge on the same conclusion. If you are making observations in different areas of reality with different technologies, different methods, and they come to the same conclusion, that's what my friend and colleague, Ed Wilson, has called consilience, the jumping together of evidence from completely independent domains of reality. If that happens, it can only happen because those things are united, not in method, not in your questions, not in your prejudices, but in the very reality that you are studying. That is the most robust kind of demonstration, and that's what we are aiming for in the new Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean. Thank you, Dan.
Thank you, Mike, for getting us started. Let me open the floor for 8 or 10 minutes of questions, please.
while I'm warming up, Mike, this is just a parenthetical comment, but I'm very interested in the concept of using the word the human past as opposed to history, which I have always thought about it as history belongs to historians. If we want to think about the human past, it doesn't just belong to people in our department. It's a very interesting phenomenon.
I completely agree with you, Dan. I was tending in that way. I called it the past, and then Noreen Turo said to me, Mike, you call it the human past, otherwise you're going to have the geologists. Sorry, geologists. But I quite agree with you, and of course your own interest in the deep past has only reinforced that. I mean, human history is something that we make, that we reconstruct from the different types of evidence that we have about the human past. And now we have the opportunity to go beyond history, which in many people's minds is limited to the written sources that survive from the last 5,000 years, to do a more comprehensive history in time, going back before the time that there were written sources, and in areas of reality where there are no written sources, whether it is the life of children, or of slaves, or of poor women, or of the ill, or of animals, which are part of a biological community. These are parts of history that have no written record, no written champion, and archaeology and archaeoscience give us this extraordinary window into the lives of all these wonderful human beings who existed and whose contribution to our world we are still building on, but we don't know it. We're not aware of it. And with this, we'll be able to recover that and understand it, I think. Scott.
Will the activities of the center focus primarily on old world European questions, or what's the geographic scope of the activities?
So I'm interested in everything, as we well know. We like to be focused and make some headway in one area. So our analytical set of questions for perhaps our first 5-year program is migration, mobility, ancient pathogens, and it's already pretty gigantic to be focusing, as we started out really kind of looking at the Aegean, and then we realized, we should probably look at the whole Eastern Mediterranean. And then we had a brainstorming session, and we're collecting samples across the whole Mediterranean. So, and Africa is of interest, so it's growing, Scott. But we're going to try and focus, at least at the outset, to do well what we're trying to do, since it's completely new, in the areas where our different expertises are strongest, but to be open and willing and eager to be a platform for others to come aboard and to lead us in new directions. Because everything's connected. It is connected. These diseases, just don't spring up in the middle of the Roman Empire. They come from somewhere else. The climate patterns that we're seeing, everything is connected. So we can seal it off and pretend we don't want to see it. We can be open to it and keep our eyes peeled. But we have to be careful, because one can spread oneself too thin. I mean, we're already doing all aspects of human existence, really, over a couple of millennia in one part of the globe. So that's a pretty tall challenge.
You mentioned the two words, Bubuan and Ingwen,
Yes.
as possibly suggesting a foreign origin for this.
According to the standard etymological dictionary, is correct. Bubon appears already in Homer. Ingwen goes pretty far back in Latin. I can't remember off the top of my head what the oldest attestation are. There is Saussure, the guy who invented, the Swiss linguist who invented structuralism, proposed a kind of, an Indo-European linguist might have a stronger opinion on this than I do, a comparison with Old Icelandic that made him think that it could possibly be Indo-European. I haven't seen anybody else who agrees with that. So both of these terms appear to come from outside the Indo-European language. Now, usually, if your civilization borrows something, a new thing, it very often borrows the word for it. So, sushi. Americans didn't eat raw fish until we bought sushi. We got sushi. Now, sushi doesn't mean raw fish. It means the rice that you have with raw fish, but close enough. So we can look at the English tongue. We have many things that we got from the Viking occupation of England. Husband, husband, your house ally. Law, that's a Viking word. Steak, steak is a Viking word. Knife is a Viking word. And so we can see different parts of reality that are borrowed, and that ancient borrowing is marked by having borrowed a foreign word to designate it. And so I asked myself, where does the term bubon come from? What language does the term Ingwen come from? And does that have some hint for us that we've never noticed about the origins of this disease that we now know existed in Homer's time? got an etymology for me?
I have a specialized question. Is there indication of bubonic plague in the Far East in these times?
So this is a big problem. We don't, I mean, the conventional wisdom is it came from China. The Justinian, the earliest evidence that has been adduced for there being this 6th century plague in China is from the 600s. So after its outbreak in the Mediterranean. So the short answer is everybody said it always comes from China. The most recent pandemic that we still have today, that started in the late 19th century and is endemic in the southwestern United States. It keeps moving closer and closer to Fort Worth every year. It's endemic among squirrels and prairie dogs. Don't pet the prairie dogs and squirrels. That started in China, and people have been thinking because of that that the source must be in China. Johannes probably can give a better answer to that than I can, but the evidence at present is quite mixed. Please, go ahead.
You mentioned something about a database about ancient shipwrecks.
Yes.
Could you tell us a little bit more about that?
Sure, sure. I was working on shipping and commerce, which is how I got interested in plague, because I learned that plague moves on ships. And so the ships are gone, but the plague's there, so you can follow from that. And I got sick of just reading sources and writing up index cards. It was really hard to count index cards, so I started making an Excel spreadsheet of shipwrecks. And then my friends at the Center for Geographic Analysis said, well, you know, why don't you put this in GIS, in a spatial analytical software, so that you can map them over time and space. And that was the beginning of the Digital Atlas geodatabase of ancient shipwrecks from 0 to 1500. We're adding now a Bronze Age component of that. It's up on the web, and everybody can download it for free and do what you want with it. Great.
Thank you.
Thank you.
And while they're setting this up, I'll introduce Professor David Wright. David received his BA from physics, which I didn't know, David, from Harvard University, and then did a PhD in zoology at the University of Oxford. And he's currently a geneticist and a professor in the Department of Genetics at the Harvard Medical School and an associate of the Broad Institute. And some of his research has involved the comparison of human DNA with that of chimpanzees, Neanderthals, and Denisovans. David.
Okay. People hear me? How do I activate this? Thank you. So I think my job is to, I think, in part to set up the other speakers who will follow me today. So I've been given the job of trying to introduce a little bit about this technology of ancient DNA, which a lot of us are using. And so a lot of the presentation I'm going to make is going to try to make the argument to you that this is a very new technology, which is not just an improvement relative to previous technologies, but rather a step change that makes it possible to do things that were not possible even 3 or 4 years ago. I think that the starting point for this, for me, is my sort of personal hero in genetics, Luca Cavalli-Sforza, who is my academic advisor's advisor, so my academic grandfather, made a bet in 1960 that it would be possible to reconstruct the history of human populations around the world based on the genetic structure of indigenous people around the world today. So that by looking at the 7,000 or so different groups in the world, assessing their genetic material, understanding how they're related to each other, and reconstructing their shared ancestors, one would be able to trace the migrations of humans around the past. And that bet that he made in 1960 seemed to be very productive and successful, and to be finding out many, many new things over the subsequent decades, as with great virtuosity and collaboration across archaeology and linguistics, he and colleagues made many claims and suggestions about what had happened in the past. But that bet has turned out to be a complete failure. And the reason is that people have moved too much. So the idea that the last 500 years were uniquely disruptive times in human history, where people have moved so much more than in the past, and that prior to that people really reflect who's been there for tens of thousands of years, has turned out not to be correct. That we know that because of ancient DNA, which has revealed that people living in each place around the world today are very different from the people who lived there 5 or 10 or 15 or 20 or 25,000 years ago, reflecting waves of movement and mixture. And it's really by only being able to get DNA from the past, from the times that matter, that we can actually see what happened. And the ability to do that now really transforms what we're able to do with DNA and really changes the picture compared to what you perhaps read about 10 years ago, because we're able to do that now. So I'm going to talk about this new scientific instrument of ancient DNA, which was to a large extent invented in Svante Pääbo's laboratory at the Max Planck Institute in Leipzig, where Johannes Krause did his graduate student work and his postdoc and contributed to a lot of this key work. And I've had the privilege of collaborating with that amazing group led by Svante Pääbo. Ancient DNA in its modern form, where we examine whole genomes, proceeds approximately as follows. You take a bone or a tooth in a clean space, which is trying to protect the sample from the person who's handling it. You drill beneath the surface of that bone in a clean room. There's the drill beneath the surface of the bone. You obtain some powder. You extract it in a clean room and try to remove the mineral content and the protein content and other contaminants in the material. And then you sequence it in one of these modern sequencers. That's the approach we use. And we do a number of post-processing steps and improvements in order to improve the yield and success of these techniques. So what I started doing beginning in 2012 or 13, 2013 really, was that I'm working very closely with the people in Leipzig, but for me it was clear that their amazing, beautiful work focused on archaic humans like Neanderthals would work just as well or better in terms of higher rates of success on people in the last 10,000 years or perhaps sometimes older than that, and that would be very powerful to be able to study large numbers of ancient individuals. So working with our colleagues in Germany, what we did is we've implemented a series of measures to make ancient DNA analysis much more efficient, to convert a much larger fraction of the samples we analyze to working data, and to make it inexpensive to assess samples on a per sample basis. And so our goal was to drive the cost down to sort of around the cost of a radiocarbon date so that we could study large numbers of samples. And this work was primarily done in collaboration with Matthias Meyer and Xiaomei Fu, who were in Leipzig at the time and who invented these techniques for enriching the DNA that we obtained from these skeletal materials for parts of the genomes that were informative and of interest. And Nadine Roland, who's the lab director here and who also was from Leipzig originally, who implemented this in our laboratory and helped us to adapt it to the particular subset of the genome we were focused on. And what we've ended up doing is enriching these samples for about 1 million positions in the genome, where many, many other samples have already been assessed, modern and ancient, so that we can compare them all to each other. And this is really very transformative because it reduces the amount of sequencing we have to do per sample by 100 to 1,000-fold, and it really makes it possible to efficiently screen large numbers of ancient samples. So this is just a comparison to a very important curve that probably a lot of you know, Moore's law, which has nothing to do with genetics, but is the rate of increase of density on integrated circuits in the computer industry, which have doubled about every 1 to 2 years over the last 75 years, and have been driven the computer revolution and a lot of the changes. The increase in ancient DNA has been much more rapid and has gone from 0 in 2010 up to a very large number now. Where there were perhaps 4 genomes or 5 genomes in 2010. There was a burst in 2014 of about 40 of them. In 2015, it kind of went into hyperdrive. And right now, there's more than 3,000 samples that, for example, we have access to in our laboratory. There's much more unpublished than published data, not because people are holding back, but just because it's taking time to bring the material to publication. So I'm going to tell you about 2 vignettes just to show you the power of the ability to look at large numbers of samples. And here I'm going to present work that was led by Joseph Lazaridis, who's going to talk next about a different project. So I'm going to talk about his project from last year as the first part of my talk. So one of the improvements that what was made possible by the ability to extract DNA using these efficient techniques combined with better information about what types of the skeleton to extract from and improved methods of more efficiently extracting the DNA, what was made possible in 2016 was this paper where we successfully got genome-wide ancient DNA from 44 individuals from the ancient Near East from before the start of farming, so from about 14,000 years ago from hunter-gatherers from present-day Israel to about 3,000 years ago, about 3,000 or 4,000 years ago in the Bronze Age. And so we compared this to data we had from Europe and several points in the Near East to try to understand what happened over time. And here is a plot where we look at about 600,000 positions in the genome, and we see how all these samples are related to each other. So these gray dots reflect present-day human samples, about 1,000 of them from the West Eurasia, which is a term we use to refer to basically Europe and the Near East, people who are genetically rather similar to each other. But when you try to see how similar the genetic data from these individuals are to each other by comparing them at their variable positions and seeing how much sharing there is between pairs of individuals, you see that individuals in Europe cluster on the left. The gray line on the left over here are all Europeans. The gray line on the right over here are all Near Easterners, with a gradient corresponding basically to a north-south gradient within each region. And the ancient samples are colored. And so we could really compare the Europeans and the Near Easterners. And what we saw is the ancient Near Easterners fell along this gradient with the southwestern Near Easterners from long ago, the hunter-gatherers and the first farmers from Israel and Jordan at the bottom here, and the Iranians at the top here, and the Armenians at the top here, reflecting the full gradient that you see today. What?
What's the coordinates on the first line?
There are principal components 1 and 2. So one of the things that we measured was if you take the 4 corners of this quadrant, these are European hunter-gatherers over here, those West European hunter-gatherers, for example, from Luxembourg. Right over there on the top are Eastern European hunter-gatherers from Karelia, or from Russian Scandinavia, or from further Eastern Europe. Right up there are Iranians, and right down here are Levantine first farmers or hunter-gatherers. The level of genetic differentiation between them, if you look at the squared frequency differences of positions in the genome, is about the same as Europeans and East Asians today. So 10,000 years ago, in Western Eurasia, there were at least 4 populations that were large populations and complex populations, each as different from each other as Europeans and East Asians are today. But with these ancient DNA data, we were able to watch what happened over time. And if you measure the degree of differentiation on the top for the oldest group of samples, it's about 0.098, which is about 0.1, which is about the same as Europeans and East Asians are today. And today, it's about 1/10 of that. What you can see is over time, it decreases. And the way it decreases is not by one of these groups being driven out and replaced by the others. But so here's 4 bars to represent these 4 very divergent ancestral populations. But over time, they fill in with groups that are mixtures of these other populations. So what happened was not one of these groups driving out all the others, but they mixed in different proportions in different places, homogenizing this region, not completely, but homogenizing this region such that these groups collapsed into each other, and you have the relative homogeneity that today characterizes West Eurasian. So if you, for example, look at what the 19th or 18th century race theorists would have said Caucasians, what they're talking about is a group which is actually a mixture of very divergent populations, so that if you were to classify what human population structure was like 10,000 years ago, it would be a classification which would not fall exactly or even maybe even close to the lines that we classify today. So that's an important lesson that comes from ancient DNA, which is that the populations of the past are not the same as the populations of the present. They're often just as different from each other, but they're not the same. And so our sense that the present-day population structure is somehow a long-term fundamental feature of human variation is actually immediately falsified even as recently as 10,000 years ago, which is a relatively short span in our history as a species. So here's a drawing that Joseph made of the collapse of these groups into each other. So the second vignette I wanted to tell you about is a work that's been led by another postdoctoral scientist in the lab, Inigo Elald, and it's about the Beaker complex, the Beaker phenomenon, and genetic changes in across Western, Southern, and Northwestern Europe associated with this phenomenon. And so The beaker phenomenon is a complex of artifacts that are not always the same in each place, but are often characterized by these bell-shaped pots. They first appear in the archaeological record probably in Iberia about 4,700 years ago, although that's a little contentious. But within 200 years, they've spread all over Western Europe. They're in Britain, in Ireland. They are in southern Italy and in Sicily. They've gone all the way east to Hungary and to Poland. It's a vast expanse, and this culture is replacing or intercalating with cultures that were much more small scale before this time. And it's always been a mystery who these people were, what drove them to spread over such a distance. And one of the things that we wanted to do with our power to look at large numbers of samples was to investigate what happened. And so this is a study which is of 400 newly reported ancient samples, which shows the power of this technology. So when we started this project, the beaker individuals we had were from Central Europe, from Germany and the Czech Republic. And now in our new data, we have them from across the range with some gaps, a few gaps, but largely across the range and covering many of the key places from the locations shown here. This is actually an out-of-date map, and it's a little richer now as well. So if you look at the same plot that I showed you before of West Eurasians, about 1,000 samples from West Eurasia today, what you see again is these two parallel lines. And I want you to focus on the left side of this line, which is just Europe. And the beaker individuals who are colored here are all on the left side. So they fall within European variation. Now, if you look today at the Europeans, at the bottom of this gradient, they're Southern Europeans and especially Sardinians. We interpret this as people who have relatively isolated descendants of the first farmers who got to Europe 8,500 years ago. And at the north side, these are Northeastern Europeans, and they are relatively higher proportion of ancestry from the Pontic-Caspian steppe due to big events that happened 4,500 years ago. And you see people in Europe are in between this. So what's very interesting is that the Iberian beakers, which is where the beaker complex first arises, are falling closely on top of the Sardinians. But the Central European beakers are all shifted toward having more ancestry from the steppe. So this was a very striking observation for us, because the other ancient cultures that we've analyzed data from, for example, the other archaeological cultures like the Yamnaya from the steppe, or the corded ware at the time that we first looked at them, or the linear band ceramic, were all genetically homogeneous. They would all fall at one place. But here we had a culture where people were diverse but had shared cultural elements. That was very exciting to us because it's a multicultural phenomenon of people who are genetically variable but have shared cultural features. And so what we are able to reconstruct from the data is the proportion of ancestry from the steppe shown in this map. And what you see is in Iberia, there's very little ancestry from the steppe, just sporadic individuals. But in Central Europe, almost everybody has ancestry from the steppe. And in fact, we're able to show that there's essentially no or very little movement between these two regions, despite the flow of similar artifacts. And so it's a spread of ideas, kind of like religion spread today, or use of technology, without substantial spread of people, which is a very interesting phenomenon and very exciting for us, because in genetics, we're used to always explaining things with movements of people, and it's very exciting to show that there's a movement of ideas that's not associated with movements of people, and this sort of enriches our understanding of these events. But after this spread, the beaker spread from Central Europe to Britain was a movement of people, and we see it as follows. So if you look at Y chromosome types, which you inherit from your father if you're a man, and his father inherits from his father, the farmers of Britain, and we have 33 males in a new time series from Britain, so we've reported in this paper also 155 individuals from Britain from about 6,000 years ago to about 2,800 years ago. And what you see is that before 4,400 years ago, they're all blue. They're all of Y chromosome types that are typical of the first farmers. And then after that point, suddenly there's almost no blue left. So this is a huge transformation in Britain. And if you look in the genome-wide data, this is what happens after the beaker phenomenon arrives in Britain. Before this point, and I'm not even showing these bars, and it would all be blue. But after this point, everybody has this red ancestry, which is consistent with being derived from people from the continent who are associated with the beaker complex. And this is not just initial, it's a permanent change. And the long-term establishment by the late Bronze Age, which is 1,600 years later, is only 90% of the population has been replaced, and it looks like that happens within a few hundred years at maximum. It's very interesting. The older population was practicing cremation, so perhaps we're not seeing them at the time of the transition, But within a few hundred years, it's hard to imagine that that population was persisting without being fully mixed in. And so it seems to be a permanent change. It's a very dramatic change achieved by this movement of people that initiates with this Beaker complex. So it's very interesting how the Beaker complex is associated both with a movement of ideas and also then a movement of people. So it's very interesting also Stonehenge, the sort of one of the great monuments of the period, is built by these farmers, these first farmers of Britain, and it begins to be built about 5,000 years ago. The Beakers arrive right in the middle, and they take it over. But it's genetically, they're very different people. So I think that's sort of the end of my introductory talk. I wanted to talk about sort of what's actually happening in the ancient DNA field, which is a lot of the talks that a lot of what I've been saying has been about events that occurred before 4,000 years ago. That's where we happen to have focused, in part because of the interest of the people who started working in this area. But there's important questions about how the past connects to the present, about how these ancient groups that are being characterized with the genetic data relate to the groups that exist today. By the Beaker period, the ancestral components in Britain, for example, are very similar to those today. So genetically, on where a present-day Briton will fall is going to be very close to where an ancient British speaker person will fall. But that doesn't mean that we British people today are directly descended from the Beaker individuals. There could have been later movements from the continent or mixtures of other people, of people who were themselves genetically similar to in their ancestral components to the Beaker groups. And so you're not going to detect them with some of these techniques that we're using. And so there are things that we can do and improvements we can try to make to track these, and there's many ideas that can be used to make progress. So I think that Joseph's talk next, which will be an example of some of this progress between 4,000 and 3,000 years ago in the Aegean region. And the conclusions I wanted to make is that ancient DNA studies have been transformative in changing our understanding of the past. The technology is going through a super exponential growth phase right now, and it makes it possible, as other scientific instruments have made it possible, to look at things that have never been looked at before. It's a huge change. It really makes it possible to look at things that have not been looked at before, and to find out things we didn't expect. And that we have an unprecedented opportunity to study the population transformations that connected the prehistoric world to the classical world to the present. And I think that that's one of the things that really is centrally going to be what we focus on collectively with the MOM initiative, where we're going to hopefully try to do this across the Mediterranean region. The technology, as you see from the first part of my talk, is very successful now, even in difficult, warm places like the Near East. And it's very conceivable that we can make progress in this enterprise. And so it's something that I look forward to working on with everybody here, and especially interacting with archaeologists, and over this period in the Mediterranean, which is a literate world over this time frame, trying to also interact in a serious way with the historians as well.
Great talk, David. I have a question about analysis. I'm wondering what sort of analytical tools are emerging that specifically capitalize on sampling multiple time points. It seems like many of the analysis you showed was robust, but not taking there's sort of one time point overlaid over the present, perhaps. I'm wondering, are there methods out there that can take advantage of the fact that we have multiple time points from the samples?
Yeah, that's a great question. And of course, it's something that evolutionary biologists studying things like bacterial time series are struggling with to some extent as well when you have temporal data. We're having to be forced by the data to try to make progress in that direction. An example that we're working on right now is trying to make progress in understanding the time course of mixture between hunter-gatherers and farmers in Europe. And we have time series data, for example, through Hungary in a paper that we have in press, I guess. And what you can see is that over time, as the radiocarbon dates approach the present, the dates of the mixture get older, but not as much older as you would expect from the radiocarbon dates getting closer to the present, indicating continuing gene flow between hunter-gatherers and farmers is going on. So this allows us to get some kind of leverage on the dynamics of the mixture rather than to just sort of make the simplifying assumption it all occurs at once, which is what we've traditionally doing before. But there's all sorts of things you can do with chronological information. You know, the most obvious one in a certain case, and maybe one of the most exciting ones, is just to figure out when particular biological innovations like lactase persistence arise. In Europe, where we have our best data, you can see that the ability to digest cow's milk, which is driven by a particular known mutation, really only gets going and rising in frequency less than 4,000 years ago. It's not really present at appreciable frequencies in any of the individuals I was talking about today. It's rising after that point, and that contradicts some of the earlier literature where people imagined it was already appreciable frequency. It was already present, but not really an appreciable frequency thousands of years earlier.
I had thought it was a great thought, very interesting. I had two questions. First, you mentioned that ancient DNA, human DNA, is difficult to do with because it's not human. The solution there was enrichment of a million sites or something like that. What I didn't understand is what enrichment, how do you do this enrichment?
The question was how to enrich for these positions in the genome. So it's a technical thing, and the methodology is used as you extract all the DNA in the sample that you can, and it's mostly microbial DNA from the bacteria and fungi that were in the soil or in the bone after the individual died, mostly, usually. What molecular biologists have done is they have techniques that hybridize the DNA mix that is produced to a set of artificially synthesized probes that are at positions of the genome that one's interested in. And basically, it sticks, what sticks to those artificially synthesized probes. So you can design and print millions of probes, and what you can use you can wash your extracted DNA over those probes, and what you're left with is highly enriched for the parts of the genome you're interested in. So it's a very powerful tool, can be used all sorts of ways. Not every analysis can be done with data like this, but it's a very powerful tool, and it makes it efficient to analyze data like this.
The second question was, you concentrated on PC1 and PC2. What can you learn from PC3, PC4, and so forth?
Yeah, the question was, these plots were principal component 1 and principal component 2. It's a good question. There's lots of other information in higher principal components, and in fact, the best inference we do is often not based on the principal component analysis at all. Those are really just used illustratively to sort of gesture at what we're doing and to wave hands at what we're doing and to hand wave about what we're doing. But the hard work of trying to make tight arguments is done not with that type of analysis, but with other analysis, which is more interpretable, we think, in terms of migrations, mixture proportions, and time, and even times or relative times than is the principal component analysis. Malcolm.
David, thank you for that really exciting summary of recent developments in Europe. And of course, we've got all sorts of new information. The recent study of burials in Wiltshire near Stonehenge and Avebury shows a tremendous influx of people at sometime between 2500 and 2300 BC from the direction of the Netherlands, perhaps replacing the builders of Stonehenge and Avebury rather as things were getting complete. And we also have the evidence, of course, of the randomized analysis of the Amesbury Archer buried in such great state near Stonehenge. Who would have guessed it? He spent most of his life on the continent. And of course, we're struggling to understand what this means. And in that connection, the dates, of course, are very important. Not least because the question arises whether this influx is somehow related to the great climate event of 2300, 2000 BC. In the UK, you have, of course, the dates come from radiocarbon dating collection. And in the UK, you have the problem of what's called the island effect in Japan, but mostly the fact that the UK is frequently enveloped in great fogs. And of course, this could bring maritime elements which don't contain the carbon-14 isotope and artificially push dates back a couple of hundred years. If the dates are really closer to 2300 BC, then people may be coming because of the great climate events. Rains have stopped everywhere else. If you're going to find rain anywhere in Europe, it's more likely to find it in the United Kingdom.
Yeah. One of the things, just riffing off with something you said, Malcolm, one of the things that's very interesting is integrating this information with the isotopic information. So isotopes measure or potentially measure migrations in a lifetime, but not the migrations of one's parents or grandparents or great-grandparents. So an interesting discussion we've had with our UK collaborators in the course of working up this study has been trying to understand how to reconcile the genetic results, which indicate such a massive population turnover in Britain over a timescale of a few hundred years, with the isotopic information of this same period, which has been argued to reflect extremely limited movement of people, less than 0.5% people moving from a place that's geologically, strontium-wise, I guess, different from the place of origin. And so what the isotopes can show is there's a tiny fraction of the individuals who have been sampled, I think order of a few hundred or even 600 individuals who have been analyzed who have a strontium movement signature. And it seems hard to believe that over a space of a small handful of generations that could be enough to achieve what we're talking about. However, the strontium isotope signature in Britain is very, very similar and undistinguishable from that in the Netherlands or northern France, and so you can't rule out these very plausible source regions for that migration. So it really is not a contradiction at all, and it's just telling you that they're not coming from places where the strontium isotope signature is very different. So I think that these two types of information together sort of provide complementary and powerful information. Isotopic information is blind or nearly blind to movement between places that have similar geological signatures, and it's blind completely to movements of one's ancestors rather than within one's lifetime. However, genetic data is blind or nearly blind to movements of people between places that are genetically very similar to each other, even if you can pick it up with strontium, and it can't tell the difference very well between movement in one generation or a movement 40 generations ago. So it's a sort of interesting phenomenon, and the two together might be interesting to consider together.
Hey there. So as someone who's getting more and more interested in this field, I have a bit of a question for you. So if you were to pose a question that you hope to see answered by ancient DNA in your lifetime, what would you say is like the one most important question? And how feasible do you think it would be to see the answer?
The most important question in ancient DNA, it's really an atlas building project from my point of view. I think that it's kind of like the invention of long distance sailing and the ability to chart coastlines, or sort of the invention of a telescope in some centuries ago. It's the ability to look at things that were never looked at before and to create an atlas and a map and really just a survey of what the landscape was like. And I think that that is the enterprise that collectively we should be engaged in, which is to map out the landscape and then to interpret and understand and hold it in our minds and reconcile it with the other types of evidence we have. That's something that's eminently possible to do now with this technology, which makes it possible to study thousands and even tens of thousands of skeletons feasibly with the resources that a group like this has and will have. And so I think it's really an exciting enterprise and will unfold over the next 5 years.
Thank you, Julie.
Yeah. I've seen a lot of people squinting, but I understand that we cannot close this shade, so I apologize about that. We shall get to work on it for future events. We're turning now to Joseph Lazaridis, who's in David Rice's lab over in the Harvard Medical School. He's a postdoctoral fellow there at his degree at UC Irvine. And you've seen some of his research already, and he's turning now, I believe, to the recent Nature paper, the 2017. You're going to give us some of the results of that?
There it is, yeah. I press this. That's the pointer. That's the advance.
Good. Okay. Thank you. Okay, so in the first part of my talk, it will be kind of a revision of what we just heard 20 minutes ago, which is the origin of West Eurasians. And this is kind of useful to have to better understand the second part of my talk, which will be the main topic, which is the genetic data that we have obtained from Minoans and Mycenaeans from the Bronze Age in Greece. And in the last part, since this is the inauguration of a new research center, I thought maybe I could give some ideas of what we could accomplish in the future. Okay, so the first part of my talk, which is the summary of what we've learned about the origin of West Eurasians, summarizes some papers that were written over the last 3 or 4 years during this great period of exponential growth that David mentioned. And okay, so if we do a PCA of present-day West Eurasians, we see that there's a differentiation between Europeans, which are shown in circles, and Near Eastern populations, which are shown in squares. along the first principal component. Along the second principal component, there's a differentiation between populations bordering the Mediterranean Sea, which are shown in blue, and non-Mediterranean population, which are shown in different colors from Europe and the Caucasus and inland parts of the Near East. And finally, there's this interesting feature of the data, which I call the bridge, which kind of bridges Europe and the Near East. So this includes populations mainly from the Mediterranean and also some Jewish groups that are also part of this bridge. Okay, so for the next part, I will show the modern populations as gray dots, so as not to confuse, and the ancient samples I will represent with colored symbols. And I will plot various ancient populations onto this scaffold to kind of show the main features of West Eurasian history as we understand it today. Okay, so the first group of populations are the hunter-gatherers of Europe, and these fall beyond Europe in the direction of Near Eastern to European differentiation. So what this is and there is some structure here in the sense that the samples from Eastern Europe are kind of on this top side of the plot, whereas the ones from mainland and Western Europe are here. So what this is telling us is that one of the reasons why Europeans are differentiated from Near Easterns is that they have absorbed some hunter-gatherer ancestry from the people that lived in Europe before the advent of farming. The second group of populations I want to show are these farmers from lots of different parts in mainland Europe, as well as Greece and Anatolia. And these populations do not overlap with modern Europeans, except with Sardinians, as David mentioned. And they have some variation in terms of their hunter-gatherer ancestry. So these samples have more hunter-gatherer ancestry, whereas the Anatolian ones have least. So what we can say is that European farmers were formed by some admixture of Anatolian and Greek farmers with hunter-gatherers that they encountered as they moved into Europe. The third set of populations are the Bronze Age people from the steppe around 5,000 years ago. And these are on the opposite side from the farmers, on the top of the PCA. So the steppe people and the farmers kind of bracket the modern Europeans. And if we look at samples from the late Neolithic to Bronze Age, down to around 3,000 years ago, we see that they are kind of intermediate between the steppe people on one side and the farmers on the other. So, and they overlap the modern Europeans. So we can say that by the Bronze Age, the main features of European genetics were formed. Okay, so let's leave Europe now and go to the Near East. And in the Near East, if we look at the late hunter-gatherers, the people that lived just before the advent of farming, and the first farmers in the Levant and in Iran, and also the Caucasus, they group into two very distinct clusters. The Levantine farmers are in the bottom part of the second principal component, whereas the Iranian and Caucasian, that's a kind of loaded term. The samples from the Caucasus are at the top of the second principal component. And as it was the case in Europe, the later samples from the Near East are also intermediate between the most extreme populations that lived there at the beginning of the Neolithic. And they also overlap the Near Eastern populations. So if we summarize the whole West Eurasian kind of history as we understand it from ancient DNA, it is in terms of this quadrangle with the extreme populations that David mentioned. And all ancient samples that we have, as well as most modern West Eurasians, fall within this quadrangle. So we think that we can explain a lot of the variation that existed and does exist in West Eurasia as a mixture of these four corner populations. So the main aspects of this difference is a differentiation between the Near Eastern and European population, dominated by principal component 1, and the population from the southwest and northeast, dominated by principal component 2. And one thing that is kind of missing from this picture is this bridge that I talked about earlier, which we currently do not have any samples from this bridge, or rather, we will have some samples from this bridge in 2 more slides. But one of the interesting things about that is that it seems to be made up of populations that were formed at different times, because obviously there has been a lot of opportunity for migration between Europe and the Near East in the ancient period with, let's say, the Roman Empire, or the colonization by Greeks and Phoenicians and so on, or other historical events. So it was always a question whether this bridge was formed in historical times, or whether there were prehistoric populations that were also intermediate between Europe and the Near East. Okay, now I'll switch to the main part of my talk, which is the study of the Minoans and Mycenaeans. And this was a collaborative effort between our lab and Johannes' lab in Germany, and also the University of Washington. So the samples that we had were mainly from the Bronze Age, and it includes samples from the mainland of Greece, which we label Mycenaean, and from Crete, which we label Minoan. And also we had some samples from people who lived in southwestern Anatolia, which are kind of the neighbors of the Aegean populations to the east. Okay, before I go to the genetic results, it's important to realize that these terms, Minoan and Mycenaean, have not always been with us. They were largely invented after the discovery of the excavation, rather, of Mycenae and Knossos in the late 19th to 20th century. And this, I'm showing some plot of their relative popularity over time. If anyone knows why Minoans were in vogue in the 30s, maybe they can tell me. I have no idea. But basically, these terms kind of describe entities of the Bronze Age more than 3,000 years ago. And one of the things that we wanted to learn in our study is whether these terms actually have some correspondence to the genetics that we see in that period. So before I go into the genetics, a little primer on the Minoans and Mycenaeans. So the Minoans are famous because they were some of the first Europeans that left written records, but their language is undeciphered, so we don't know what language they spoke or how it is related to other languages of Eurasia. In the case of the Mycenaeans, their language, which was written in the Linear B script, has been deciphered since the 50s, and it is known to be an early form of Greek. So this ties the Mycenaeans both to later speakers of the Greek language from antiquity down to the modern times, and also to the other speakers of Indo-European languages across Eurasia. So we kind of have some idea of who the Mycenaeans might be related to, but the Minoans are kind of an unknown quantity. And one of the other things that we wanted to know with this study is whether these populations that apparently spoke different languages, whether they were genetically similar or quite different. And so the result is that they are quite similar. That's the main finding of this study. And also they fall on this bridge that I talked about earlier. So the Minoans and Mycenaeans, as well as the Bronze Age Anatolians, they are, in the context of West Eurasia as a whole, quite similar to each other. Another thing is that compared to the earlier farmers from Greece and Anatolia, they have this shift to the top, towards mainly the direction of the Caucasus and Armenia. So if we zoom into the Minoans and Mycenaeans samples, we see that the Mycenaean samples cluster to each other and they're more towards the European side. The Minoan samples also cluster with each other and they're more towards the Near Eastern side. The Bronze Age Anatolians also cluster with each other. And so overall, we can say that these labels that we assign to this data, Minoan and Mycenaean, they have some correspondence to the genetic makeup of these individuals. But things must have been a little more nuanced because, for example, we have this sample from Armeni in Crete, which postdates, it's around from the 1300s BC, and it postdates the arrival of Mycenaean culture on the island of Crete, and it is actually more extreme than even the Mycenaean towards Europe. So maybe there was some undetected population structure that existed in Crete, or maybe this was a migrant from the mainland. So basically, I want to urge some caution that this structure that we see in the samples that we do have may not reflect the full kind of population structure that existed in the Aegean during the Bronze Age. Okay, so we can also model the ancestry of these different populations as mixtures of other populations. And so I use different sources from Eastern Europe, Iran and the Caucasus, the farmers of Anatolia, and the farmers of the Levant. And what we can see is that all the groups can be modeled in different ways as mixtures mainly of the early farmers of Anatolia and Greece, who are also quite similar to each other, represented by this green color. All the groups also had some ancestry related to the populations of Iran and the Caucasus, which is represented by this red color. But whereas this was sufficient to model the Minoans from two different sites in Crete, it was not sufficient to model either the Mycenaeans, who seem to have some extra ancestry related to Eastern Europe and Siberia, represented in blue, or the Bronze Age Anatolians that seem to have some extra ancestry related to the Levant. So overall, we can say that all these populations are very similar to each other, and they're made up primarily of Anatolian farmer and Iran Caucasus-related ancestry, but there were also some minor differences. We can also try to find more close sources to these populations. So the ancestry that I was talking about from Eastern Europe or Siberia or Iran, it did not arrive in the Aegean on a flight from Novosibirsk, for example. So we can search the different populations that we have currently sampled and see whether some of these populations could be candidates for the more proximate source of ancestry. And when we do this in the case of the Mycenaeans, we see that there are two alternative models that fit. So we cannot resolve which of the models represents the true history. And indeed, the true history might be something more complicated, relating to populations that we don't have any samples from. But these are our best guesses. So the Eastern model models the Mycenaeans as a mixture of the local Anatolian farmers and a population related to ancient Armenia. And the Northern model represents the Mycenaeans as a mixture of a population like the Minoans and some ancestry related to populations from the steppe. Showing on the PCA, we see that these are the Mycenaeans, and you can model them as either this Eastern model, Anatolia plus Armenia, or the Northern model, which is Minoan plus steppe. And geographically, we can interpret this as follows, if we kind of made a scenario. First, you have a settlement of both mainland Greece and Crete from farmers related to Anatolia. Then you have some movement of Iran Caucasus-related ancestry that goes only into the island of Crete. And finally, you have some ancestry related to populations from Armenia that goes only into Mycenaeans. So that's the Eastern model, a caricature of the Eastern model. In the Northern model, again, you start by having Anatolia-related ancestry in both mainland Greece and Crete, followed by movement of this Iran-Caucasus ancestry into both mainland Greece and Crete, followed by some steppe-related ancestry that reaches only the mainland and doesn't make the jump into Crete. So as we sample more and more populations from both areas north of Greece and east of Greece and so on, we might be able to resolve these two models and see exactly what happened. Another thing I want to point out is that these arrows that we saw here, it doesn't necessarily mean that there's one big migration taking place from the steppe to Greece, let's say. It could be some gradual arrival of migrants over a long period of time that eventually makes the mainland somewhat genetically differentiated from Crete. Okay, another interesting thing that we can say about the Mycenaeans is that we can compare them against modern populations. And when we do this using FST, which is a measure of genetic distance between the Mycenaeans and different populations, we see that the lowest differences are in the Aegean and in surrounding areas. So we can say that of the modern populations from southeastern Europe and Italy and Cyprus and so on, they still retain some affinity to the Mycenaeans that lived there during the Bronze Age. And this is kind of a general pattern that we see in different parts of Eurasia. For example, there's a different paper that was recently published that showed that the modern Lebanese, they have some affinity to the Bronze Age people of Lebanon and so on. Another thing that we can kind of say with this data is to infer some phenotypic traits. And the traits that are most well understood right now are probably those having to do with physical appearance. So this is like a kind of a set of pictures of art from the period. And if we predict some phenotypic traits of hair and eye color of the different groups that we have, we see that they had a lot of variation, so they had kind of many of the extremes of phenotypic variation, but overall the model, let's say, appearance was had a brown shade of hair and brown eyes, which is kind of corresponds to what is depicted in the visual art as well. Okay, so the last and brief section of my talk is a couple of ideas on what we can do in the future. So, in this slide, I'm showing all the Bronze Age populations from the Mediterranean that we currently have. So, there's a very distinct pattern where you start from Iberia to the Aegean, to Anatolia, and to the Levant, and which is a very clear west to east pattern. So, one thing that I think we should work towards is to kind of have a complete inventory of populations all across the Mediterranean to see exactly where they fall on this line. And this will be useful both in itself, but also as a kind of tool to be able to understand later population movements. For example, if we know what kind of people lived across the Mediterranean, we can later understand what the impact, let's say, of the sea peoples on the Levant were, or the Phoenician colonies in the Western Mediterranean, or the Greek colonies, and so on. The second thing I want to highlight is that if we compare the ancient samples that we see in different parts of the Mediterranean with modern people that live there, we see this very interesting pattern that all the modern populations, they are related to the ancient ones, but there's a systematic shift to the north, in the sense that they have acquired some ancestry from outside the Mediterranean. And this might be important because it kind of tells us that as we study the Mediterranean, we cannot study it in isolation, but we have to figure out exactly how the Mediterranean is affected from other parts of Eurasia, like mainland Europe or the Caucasus, and so on. And with that, I'd like to thank you and take questions.
Thank you, Joseph. If I could actually lead off with a question, I'm quite curious to know more about the sites from which the samples are coming. For example, if they're coming from burials, and if there's differential access to burials, elites, non-elites, are we sampling elite populations here?
Yeah, so we have some information on all the samples on what the burial context is, and in the case of the Mycenaeans, one of those 5 dots, which actually I don't remember which one, comes from an elite burial. So, and it appears to be similar to the other, let's say, the people that were buried in a common type of burial. So obviously that this doesn't disprove that the elites were genetically differentiated from the common people, but it's the beginning of some kind of understanding of social structure in the Aegean. Yeah.
Not showing anything because there are no data, but it's just
Yeah, so
where do you expect them to fit onto this plot, and how does that affect the interpretation?
yeah, so in my talk in February, I kind of wished for some data from Africa, and it turned out that my wish came true because Johannes's group published some data from Egypt, and these samples from Egypt seem to be related to the populations of the Levant. And there's also some preprint that has appeared on the bioarchive that looks at early farmers from Morocco, which also seems to be related to the Levantine population. So it's important to kind of, the Mediterranean obviously has a northern and a southern end, and I think we're beginning to understand the southern end, but I didn't talk about this in this talk.
You said a little bit about data robustness. If you have a sample of 5 for the Mycenaeans, what is your expectation in the longer run if you were to guess?
Yeah.
Imagine that they would still cluster in that same general.
Yes, so actually we have a sample of 4, and an important thing to note is that a sample in genetics is kind of different from what we could think of as a sample of height or weight or some other physical property. And the reason is that each of us is made up of 2 parents, 4 grandparents, and so on. So when we're sampling an individual, we're not actually sampling one individual, but the whole number of individuals that were ancestral to that individual. To give you an example, if one of those Mycenaeans had some kind of ancestry that was atypical of the group as a whole, let's say it had one great-grandparent that came from Iran, let's say, then we would be able to detect that. So even though our sample is 4, we are kind of confident in that sample for the reason I explained, and also because these samples come from different parts of the Peloponnese and also from different social status, like I mentioned earlier. So obviously, as we sample more and more, we will see more structure, but we're fairly confident that the structure we see is not some artifact of sampling size.
Hey there, thanks for the talk.
Mm-hmm.
So my question has to do with something you briefly touched on. Your work with the Minoans and the Mycenaeans, what is that kind of revealing about the identity of the sea peoples?
The sea peoples. So that's a very interesting question. And so I think the sea peoples, as they're understood by archaeologists, they are named. So they have these various names like Sardana and so on that may be mapped to known places in the ancient Mediterranean. But the problem we have now is that, okay, now we have some fix on the Bronze Age Aegean. So if we, let's say, we sample people from the Levant and they seem to have some ancestry related to these people, maybe we can infer some movement, let's say, of sea peoples from the Aegean. But we cannot really understand the movement of the sea peoples until we have a full inventory of, let's say, ancient samples from across the Mediterranean, so we can prove that, let's say, the sea peoples originated here or there or somewhere else.
I'm just going to jump to Judy in the front, Henry, because it's directly on this point.
Just on this, I mean, you have to be aware that the term sea people is an invention of the 19th century, and it would be very hard to discover the past and invention of the 19th century. So I mean, so that shouldn't be our aim. What sea peoples basically terms is that we see an enormous amount of human mobility that starts in the 13th century and accelerates around 1200, which led groups of different origin, maybe part from central Mediterranean, like Italy, from Greece, from other parts, also from Anatolia, to move all around the Eastern Mediterranean. This makes our picture much more complicated, and I think what Joseph has already shown is now we have the possibility to even trace in individuals different kind of admixtures to see where they came from. And I think the question is not to show the sea people, because this is just the wrong question, but to say what was the mobility of these different groups all around the Eastern Mediterranean, where did they come from and how did they admix. So I would rephrase it and not try to find this, but really these tools give us the possibility to go far beyond this kind of searching for an entity that never existed.
Yeah, I just have a question about when you put up the map that showed relationships between the ancient DNA and the modern populations, there was a sort of concentric circles almost of the color getting less and less similar, except for southern Italy, which was this like deep blue.
Yeah, so actually FST is affected by genetic drift. So if a population has a small effective population size, it can be highly drifted. So that would cause it to be to appear to be highly differentiated from a population of interest. So there are other measures of affinity that we can test that are not affected by this phenomenon. I don't think there's anything particularly weird going on in southern Italy.
I just want to ask a hypothetical question. If you had a cemetery which had 1,000 individuals in it, and you had the money to sample each one of those 1,000 individuals, what sort of result do you think you might get, which would be presumably very interesting and perhaps more instructive than just sampling 5 individuals?
Yeah, so I think with there's lots of things you can do with large numbers, but one thing that is clear about this period is that it's international in nature. So you see artifacts from the Aegean turning up in quite diverse contexts all over the Mediterranean and even in Northern Europe. So there must have been some trade network or something that linked the Aegean to different parts of the ancient world. So with 1,000 individuals, I would predict that perhaps we would see some foreigners, foreign traders or something that might appear in the Aegean. And yeah, I hope someone gives us the money to sample all those individuals.
Joseph, thank you very much. First, with regard to the Sea Peoples, the August Austrian Academy held a major conference about 2 years ago. The papers have just been published within the last couple of months devoted to the collapse of civilizations at the end of the Bronze Age and the Sea Peoples. The papers are well worth reading. Of course, you can start with mine. But it's a complex phenomenon because we also had the collapse of the Egyptian New Kingdom, the Hittite Empire, Mycenaean Greece, tremendous movements of peoples. On the matter of the general population of Crete and the Mycenaean mainland, we have now, of course, also the DNA evidence from animals. And one very interesting study was published showing that the famous sheep, and especially the Grimi of Crete, were closely related to those of the Gasulian culture of the 4th millennium BC and centered in what is now modern Israel, which implies that a group for one region or another made its way up the coast, say to southern Anatolia, and then came across at that period. Curiously, a former student of mine, Professor Bobby Cole at Hunter College, who's a leading Minoanist, spent a summer, I think when he was in high school, working in excavation in Israel, and suggested years ago that the sort of hutterns, these small burial huts made of terracotta, their decoration in Israel resemble things you'd seen maybe 500 years later in Crete. Everybody sort of smiled and said, "Sure, Bobby," and patted him on the head. But with this new information from the DNA studies of animals, it begins to look like a good idea.
So just one brief answer. So I think, so in this study we kind of didn't see any influence from the Levant in the human populations of Crete. But of course, going back to the earlier question, if we had a larger sample size, maybe we can see some human correlate of this possible movement of sheep into Crete.
I think there might have been one last question in the back, perhaps. Lisa, was there someone? No. Good. Okay. Thank you. Thank you. I would like to introduce and turn the floor over to my colleague Joyce Chaplin, Professor Joyce Chaplin, who's a colleague of mine in the history department. I'm afraid I have to run in class, so Joyce will be taking over the chairing for the remaining 2 panels, beginning with Johannes Krause. Okay.
Well, it's my very great pleasure to join this event to introduce Johannes Krause and moderate questions. Johannes is based at, and I've forgotten what your preferred pronunciation, if it's MAM or not, MAM, Max Planck Institute for the Science of Human History, and he will be speaking to us now on genomic evidence for prehistoric pandemics.
The microphone doesn't work. I can also stay here. The microphone doesn't work? No. Does work? Okay, perfect. So I would like to take this chance today to talk a bit about our ancient pathogen research. As many of you have heard, this is something that our lab has been quite involved in over the last couple of years. And in particular, I would like to show you how we use genomic research to provide more evidence for historic as well as prehistoric pandemics. So today it is believed that the history of infectious diseases is really starting at the beginning of the Neolithic, because that's the time when humans also come into close contact with animals. Probably a bit more than before because they start to domesticate them, and it is believed today that due to this domestication, pathogens actually enter the human population in the process that is called the zoonotic transmission. And the evidence comes mostly from the fact that when we look at the closest relatives of those infectious diseases, today we find their closest relatives in those animal populations. So there are examples such as measles, smallpox, flu, tuberculosis, plague, leprosy, or pertussis, where we find the closest relatives in animals. And of course, also humans move into closer living quarters. They live much closer together, and by that provide the perfect breeding ground for infectious diseases. You can imagine people living in such a settlement have a much higher chance than to get into contact with those pathogens, and those pathogens find perfect conditions to spread extremely fast. And then as soon as human populations become bigger, some of those pathogens cause large outbreaks. Those outbreaks then we call pandemics, and there are some examples that we pick up in historical records, such as the plague of Athens in the 5th century BC, that had a strong impact on the Peloponnese War. Actually, Pericles died of it even. The Antonine Plague, the 2nd century AD in the Roman Empire. Some people think that it stopped the expansion of the Roman Empire. The Justinianic Plague, we heard this morning, or also this afternoon already, was a very big event in the 6th century. Some people think it was one of the causative events of the end of the, or causing the end of the Roman Empire. And Mike is actually one of the experts on that who's written a whole lot about that. And then the 14th century Black Death, which is probably the most famous pandemic that we know of from history, and probably killed about half of the European population in the middle of the 14th century in only about 5 years. For some of those historical outbreaks, we have some idea what might have been the causative agents, what might have been the pathogens that were actually responsible for that outbreaks, but often we have actually no idea, because just based on the historical records, it's very hard to actually then know what type of pathogen might have been present in the past. But as I will show you now with molecular evidence for some of those larger historical outbreaks, we have a good understanding of what pathogens might have actually been responsible for those. And even though this all sounds a bit like it's only relevant for the past, and actually in the mid-19th century there were even a lot of medical researchers that assumed that infectious diseases should not be so much in the focus of medical research anymore, because people thought because of vaccination programs and antibiotics, we will eradicate all infectious diseases by the end of the 20th century. Of course, we know now today that unfortunately those people had been wrong. And in fact, we see since the 1980s, we see newly emerging infectious diseases, emerging infectious diseases that were not known to humanity before. Examples such as HIV, SARS, Hanta, Ebola, Lyme disease, just some examples that many of you have followed over the last couple of years that are emerging due to largely a globalized world, but also due to the fact that the human population is growing on an exponential scale. The other problem that we now have is that some of these infectious diseases that were actually thought to be under control are coming back. And that is mostly due to the emergence of antibiotic resistance. So something like multi-drug resistant TB is becoming more and more of a problem, and there are millions of people that get infected every year. So this seems to be a very interesting, therefore, topic to look into infectious diseases. But surprisingly, when we started to kind of look into that, I figured that there's very little known about the evolution of those infectious diseases, about the causative agents of those diseases. And that is largely due to the fact that when you want to study evolution, usually you have to look at fossils. And this is very difficult because pathogens usually don't leave fossils. You don't have fossil bacteria. You might have a little kind of round shape or kind of rod-like shape in stone or maybe in a skeleton, but it doesn't really tell you anything about the biology of those pathogens in the past. And therefore, we know quite little about the early evolution, about the zoonotic transmission of diseases. How did they adapt to humans as a new host, for example? What's their evolutionary rate? So what's the mutation rate of pathogens? How fast do they change through time? And that's, of course, all information that would be quite important to have. So this is then when we then decided to invent this new field of research that we call ancient pathogen genomics, where we do some sort of time travel. We go back in the past, take those ancient skeletons, as you have heard, something that we can do for ancient human genomes, we can now also do for ancient pathogen genomes. And that is quite exciting because we are basically creating some sort of fossil record, molecular fossil record of pathogens to tell us something about how fast do bacterial genomes change through time, what's their mutation rate, how did those pathogens adapt to humans, how did human populations maybe adapt to the pathogens, and then, of course, also providing information to historians and archaeologists about the causative agents of past pandemics. So what has actually caused those large outbreaks, such as the Black Death, the Justinianic Plague, Plague of Athens, and so forth. And over the last few years, we have actually been quite successful with this approach. There are a number of studies where we looked at different infectious diseases, such as, for example, plague from material from the Black Death pandemic in London. We also looked at Mycobacterium leprae from medieval time, where we reconstructed entire genomes to tell us something about the evolution of this Mycobacterium leprae. We looked at tuberculosis from the New World, which was actually quite exciting because it wasn't really known what type of tuberculosis was found in the New World, because today you find in the New World only European tuberculosis, and it was already present before contact based on archaeology and anthropology. What we could actually show was that indeed there was tuberculosis there in the New World, but it was not a human-adapted version. It was actually zoonotic transmission from sea mammals to humans, which was quite exciting to see. We've also recently not just looked at pathogens, but also commensals, so bacteria that live in you and on you, such as Heliobacter pylori that lives in the stomach that can cause ulcer, for example, and cancer. And that is actually something that we reconstructed from the stomach content of the Iceman. So from this ice mummy that was stuck in the glaciers in the Alps for about 5,000 years, we could actually reconstruct the genome of this Heliobacter pylori from this individuals to learn something about the evolution of Heliobacter pylori in Europe. Recently, a study which is still in review, a collaboration also with people from the Science of the Human Past Initiative, so Noreen to Ross, which unfortunately couldn't be here today. We did a collaboration where we looked at early epidemic burials from the contact period, actually from Mexico, from the 16th century, where we had a large mass burial and were able to reconstruct the pathogen that likely killed all those people in this mass burial, which is quite interesting because it's during the time of the Cocolitzli, which was a large pandemic probably that happened in Central America. And what we could reconstruct was actually Salmonella enterica, the causative agent of typhoid fever, which many of us had actually not on the radar to play an important role for larger outbreaks in the past, something that now that we're looking through larger data sets, we actually see quite present in also prehistory, which is quite exciting. And we have also looked at pathogens, not just from humans, but also from plants. So together with another Max Planck Institute in Tübingen in Germany, we were able to reconstruct the genome of Phytophthora infestans, which is the causative agent of potato blight that caused the Irish potato famine. We were actually able, from leaves that were dried in herbaria collections over 150 years, to reconstruct the exact strain that caused the Irish potato famine in the 19th century, and could then actually show that it has disappeared today and was actually replaced by other strains because the potatoes actually adapted through time, or basically agriculturalists kind of bred them in the way that they were adapted, which was also quite exciting. So in each of those topics, I could probably talk for an hour, so I'm trying to kind of restrain myself and only focus on one, and that is Yersinia pestis. And why Yersinia pestis? Because I think, as we've heard today already, Yersinia pestis is probably the pathogen that has the highest potential to cause massive amounts of death and destruction in the past, and might have not just played an important role in historical times, but maybe even in prehistorical times as I hope I can show you today. So a few words about Yersinia pestis. It's actually not a human disease. It's not adapted necessarily to humans. It's actually a rodent disease that is found in sylvatic rodents, so rodent populations that you find in the environment, such as squirrels, marmots, gerbils, and other rodents. And it can then actually be transmitted from those wild rodent populations into rodents that live in close association with humans, such as rats and mice, for example. And this host jump is actually quite literate because it jumps by flea transmission. And what happens is that fleas bite infected individuals, and those fleas, they take up the bacteria in their stomach. And those bacteria are actually quite nifty because they're building some sort of biofilm in the stomach of the flea so that the flea cannot swallow the blood. So what happens is the flea is actually starving, and it keeps on biting and bites individuals. And with every bite, it actually injects bacteria into this newly bitten individual, by that basically disseminating the disease. Rats and mice are actually also quite sensitive to the disease. They also die of the disease. So if there's a large outbreak within the rat population that lives in close proximity to humans, the fleas look for the new kind of host, the next warm body, basically. And if that warm body happens to be a human, then humans can also get infected. What then happens is the bacteria enter the kind of human body through the lymph system to this, what we've heard today already, the buboes, this kind of famous swollen then lymph nodes. And from those buboes, the bacteria can disseminate in the whole body and through the lymph system, then through the blood system. And from this bubonic plague, from this large buboes, the next stage then is what we call septicemic plague. So some sort of sepsis. The bacteria enter the human bloodstream and cause sepsis. And after 5 to 10 days, in about 60% of all the cases, they actually cause death. So it's one of the most virulent and mortal, or with the highest mortality, bacterium that we know. There's another form of plague that is called the pneumonic plague. That is actually the same bacterium causing the same disease, but the bacteria don't enter the body through biting or through the bloodstream, but through droplet infection when you inhale it into your lung. And that's actually a quite severe form because symptoms usually occur after about 15 to 20 hours, and death can occur after 24 to 30 hours. And that is quite severe because even today, if you inhale the bacteria, it might actually be too late for antibiotic administration. So that is actually a big problem. So the mortality for pneumonic plague even today is quite high. So plague we still find in the world today. Fortunately, it's not a big health threat anymore. We have only a few thousand cases in human populations today. Of course, it's found in many rodent populations, but it's found on basically almost all continents except of Europe and Australia. But it's also found, for example, here in the United States. So I think Mike was mentioning that already earlier. So if you kind of go in the Grand Canyon, for example, and you see warning signs that warn you from petting those squirrels and marmots, please take that serious because indeed they carry Yersinia pestis. And there's a couple of cases every year where people in the United States actually get infected with Yersinia pestis, so with some sort of bubonic form. But again, we have antibiotics today, so the treatment is quite successful, at least if you give the antibiotic in time. What we do know based on historical records as well as now molecular evidence is that there were at least 3 large outbreaks in historical time that were caused by Yersinia pestis. So those 3 pandemics, the first one starting in the 6th century, the Justinianic plague affecting mostly the Roman Empire, and as I mentioned, maybe even causing the collapse of the Roman Empire, one of the causative events. That lasted about the 8th century, and then it disappears in Europe. We're not quite sure why it disappears, but we don't have any evidence in the historical records for plague appearing again until then the 14th century. And in the 14th century, that's the infamous Black Death. In only 5 years, from 1348 to 1352, about half of the European population dies because of that disease, which is quite incredible. And then we see outbreaks of plague, many dozens of outbreaks all over Europe occurring in the next about 350 years until the mid-18th century, when again the plague disappears, in Europe at least, for good for the time. But then it reappears in the 19th century with the 3rd pandemic, where some people say it's actually still ongoing. This is the Hong Kong plague, also so-called. This is also the time when this gentleman up here in the corner discovers the bacteria which are responsible for the disease that then also gave, or he also then gave its name to the bacteria, so Yersinia pestis. And this is, as I said, maybe still ongoing or kind of not so much found today anymore within at least human populations. But it's also the time when the disease gets disseminated all over the world. So this is the reason you find it in the Grand Canyon today, or in Africa and other parts of the world, because we have a globalized world at the beginning of the 20th century when the disease can then really spread with ships and ship rats all over the world. But now, of course, you might ask the question, how can we extract, how can we then reconstruct those ancient pathogen genomes from the past? And this is indeed quite difficult, because often we do not know what a person in the past has died of. Sometimes we can use clues. For example, we had a clue about the Black Death because we had historical records that indicated that in the city of London here in the eastern part, this is the Tower of London, there was a mass burial site which is called the East Smithfield, and that was actually used only during the Black Death, and we could assume that all the bodies that were present here in this site that was rediscovered in the 1980s were from the Black Death. So we could look into those bodies and then hopefully find evidence of an ancient pathogen. In this case, it was Yersinia pestis. We found about half of our samples being positive using traditional PCR methods. So we could have stopped at this point with our project. We could have said, OK, Yersinia pestis was present in the Black Death, likely the causative agent. We don't need to go further. But as I mentioned before, we wanted to go one step further because we wanted to study the evolution of the pathogen. But how do you now get to this ancient DNA? How can you now reconstruct those ancient genomes? And as David has already mentioned, it's actually quite challenging, because if you take DNA from an ancient skeleton, the vast majority of that DNA is actually not from the human itself. It's also not of the pathogen that has killed that person, but it's from the environment. It's bacteria, it's plants, it's fungi, it's all kind of DNA that you would find in soil. So what we somehow have to do is we have to fish out the DNA that we're interested in. And that is something that we can do, as David has mentioned, with those capture approaches. For example, using a capture array, which is a little piece of glass. On the surface of the glass, we have DNA. That DNA you can synthesize. And DNA has the wonderful property that complementary fragments will bind to each other. So on the surface of this piece of glass, we have modern Yersinia pestis DNA, which you can then incubate with the ancient DNA. The complementary fragments will bind to the surface of the piece of glass, and you can wash away the background, and by that enrich for your pestis DNA. That works actually quite well. So you see that the amount of Yersinia pestis DNA in our samples from London was very low back in the day, but after this enrichment, it actually became quite high. And it was then high enough for us to reconstruct an entire genome of this ancient Yersinia pestis strain with pretty good quality. So we have seen every base about 30 times, which means it's a really high-quality ancient genome. What you can now do is you can plug these ancient genomes into the phylogeny, so into the family tree of modern Yersinia pestis. So this is basically the family tree of modern Yersinia pestis. You can see that this is about where 80% of all modern strains fall in that you find in rodent populations in different parts of the world. They're called branch 1, 2, 3, 4. And then you have, this is called branch 0. And here this is the outgroup. So this is the closest relative of Yersinia pestis, which is called Yersinia pseudotuberculosis. When you then had our first genome from the past, from London, from the Black Death, and we plugged that into our phylogeny here, we were surprised to find it at this position here, very close to the common ancestor of most of the strains that you find in the world today. And this makes maybe sense, because it was basically the Black Death that disseminated this disease all over Eurasia and gave rise to those different strains that are mostly found in the world today. What we could also show in our research over the last few years, reconstructing more ancient pathogen genome of Yersinia pestis from different outbreaks in Europe, so from the 15th century, 16th century, up to the 18th century, that after the Black Death, there was a European post-Black Death branch, or basically population that was established in Europe. So what happened is the plague came to Europe in the 14th century and established a population of plague here for about 400 years, until it disappeared again in the 18th century. So it was not reintroduced multiple times, but there was a population here, which basically tells us there was a reservoir population. There was probably an animal population, a rodent population, that was harboring Yersinia pestis for about 400 years, which is quite exciting. We don't know which one it is. There's a lot of speculation. Could be marmots, could be squirrels. We're not quite sure, but it's quite interesting to see that. What we could also show in our research is that we have multiple genomes from multiple sites of the Black Death pandemic, so of the mid-14th century, from Barcelona, from southern France, from Germany, from London, and all of those genomes are genetically identical. So all those 50 million people died of the same genetic clone, of the same bacterial clone, basically, which is also quite exciting to actually see how powerful this clone, this bacterial strain was. We don't find a single mutation in the genome of those bacteria for this 5-year period. What we also now have is the Justinianic plague, where we have ancient genomes, and they actually surprisingly fall on a dead end. So it's like some sort of cul-de-sac here. You'd actually see those genomes have their own little branch, but they didn't continue, so there's no descendants of that strain found in the world today. So we seem to have a pretty good understanding of the evolution of Yersinia pestis from about the 6th century through kind of the next 1,500 years or so. But our big question then was, over the last few years, what was happening before? Did some of those earlier pandemics be caused by Yersinia pestis, or could we actually trace it back into prehistory? And as we've heard already earlier today, this is actually something that we and others managed. But this is much more of a challenge, because you don't want to be blind if you look into prehistory. For the historical time periods, we have historical clues. We have historians that tell us there's this mass burial from this time period, there's this and the symptoms that have been described by early historians that indicate maybe it was measles, it was smallpox, it was plague or something else. But really, we don't have that for prehistory. So how can we address that? How can we make sure that we're not blinding ourselves by only fishing for a certain pathogen? How can we look at everything that was there? And the best approach that we came up with now, and that we're working on quite extensively right now, is what we call a shotgun approach. So where we're not just fishing out the DNA fragments that we're interested in, but we actually look for everything. We try to assign every DNA fragment that we get from an ancient skeleton to a certain species. What bacterium, what pathogen might it be coming from? And that is an approach that, again, 2, 3, 4 years ago would have been not possible, because it would have taken too much time. Because we were producing with those machines, with those sequences, billions of DNA fragments, and even the fastest algorithms a few years ago would have taken years for such a large data set to be analyzed. But now we have actually better algorithms, and one of the group leaders in our department, Alexander Herbig, together with Daniel Husson from the University of Tübingen, have been working hard on this algorithm that is called MALT, which means Magen Alignment Tool. I don't want to kind of bore you here with the details, but it's a very, very fast alignment algorithm that allows you to assign what species a certain DNA fragment comes to in very high throughput. So you compare to a database, and that database can be all the bacterial genomes that are known. And it's super fast, so it can assign about 1 billion DNA reads in about 24 hours. And again, this is much, much faster than what was available before. Before, it would have really taken years to do that, and we can do that now in very short time. And actually, one of our postdocs just used this algorithm to screen a data set of about 3,000 human shotgun data sets that we had available in our lab, or some of them were also shared by David Rice's lab with us. And we actually did find about 132 pathogens in this large data set. Now, this is not so much, but still much more than we would have been able to find in a targeted approach where we would have really fished in the dark. And you can see those range from kind of prehistoric samples from the Paleolithic until about pre- or post-Columbian populations in the New World. The type of data that you get out of that kind of looks like that. Now, this is maybe hard to understand. I don't want to want you to follow that in detail, but basically every column here is one individual, and every row here is a different bacterial pathogen. And what you see here with the different colors is the more red is the more positive, the more blue is more kind of weak candidate. And the most exciting thing that we found in here was that there was a whole cluster in the late Neolithic Bronze Age individuals that showed evidence of Yersinia pestis. And that was really something, for that time period, was kind of unexpected. Fortunately, in the same year, when we started to do these studies, there was a study published by our colleagues from Copenhagen, Rasmussen et al. They did find Yersinia pestis genomes at that same time period in the late Neolithic and Bronze Age from individuals from the Altai Mountains, so actually quite far east in Central Asia or southern Siberia. And we now use the individuals that we found to be positive to also reconstruct ancient pathogen genomes. And this is just the kind of radiocarbon dates that you have here for the individuals where we could reconstruct entire Yersinia pestis genomes in prehistoric time, which was very, very exciting because none of us had thought like 3 years ago that Yersinia pestis was already present in the Bronze Age or in the late Neolithic in Eurasia. We can now again reconstruct the family tree of the ancient Yersinia pestis from the Bronze Age or the kind of then late Stone Age. And when we do that, we find that they all fall on this kind of branch here, which is actually not really part of the family tree. It's more like a close related cousin, if you want, or kind of a sister clade of the modern Yersinia pestis. And what is also quite striking here is the numbers actually correspond to the age, where 1 is the oldest and 7 is the youngest. And you actually see they're like on pearls on a line. You can really see some sort of evolution going on of this strain through time, through about 1,500 years of history of this specific Yersinia pestis strain. What we could also do is, because we have now dates, radiocarbon dates of those strains very close to the common ancestor, we could actually date when the common ancestor of all Yersinia pestis and the Stone Age Yersinia pestis existed in the past, and that is about 5,500 years ago. So it seems that the divergence of those strains happened 5,500 years ago, so roughly when this disease might have entered mammalian populations. What you can now also do is you can actually link them up, because they're like nicely arranged on this branch here with arrows in space. And what you then actually see is that there's quite an interesting story, because you can see that 1 gives rise to some common ancestor with 2 here, that gives rise to 3 and to 4 and to 5 and to 6. So there is a bit of a story, actually. It looks a bit like a line through space where those genomes are connected. But then there's something really odd happening, that 6, which is actually in southern Germany in the Augsburg region, is closely related to 7, which is in the Altai region. So we would have to draw an arrow that would roughly go like that. So this looks a bit strange. It kind of looks like a weird pattern. But the human geneticists in the room, they actually have seen this pattern before, because that's exactly the same pattern that we see, and David has mentioned that, and also Josef has mentioned that, with this kind of early Bronze Age migration of the Yamnaya population going into the west as well as into the east. And then we see actually a backflow of human genetic material into Central Asia. So it's actually quite striking how much the kind of phylogenetic tree of Yersinia pestis resembles the population movements that people have reconstructed on human DNA. So there seems to be some close resemblance about plague following human migrations. What is also interesting is that the Stone Age plague is actually not identical in its genome to modern plague. It actually lacks a large region of its genome. And that's an interesting region. So this is the modern genome here in gray. This is the ancient genomes from the Stone Age. They don't have the genes that are necessary for bubonic plague. This strain from the Stone Age is actually not adapted to live in fleas and be transmitted by fleas into humans. So that is quite puzzling, because it basically rules out that this strain has been a bubonic form. It suggests that it was somehow transmitted septicemic or pneumonic. Now, pneumonic plague is very rare in the 20th century. So it's really hard for us currently to understand how this disease in the Stone Age was transmitted. We find it in high frequency in about 10% of the individuals that have been analyzed for this time period in certain regions. So it was pretty common. And it's quite puzzling. We really have no clue right now how this disease was disseminated. And then at the very end, so just in summary then, what I showed you is that Yersinia pestis has caused at least 3 historical outbreaks, pandemics. There was a high mobility of Yersinia pestis through human history, so it seemed to have moved somehow in connection with people. It might have caused already larger outbreaks, such as pandemics or large epidemics in prehistory, so in the Bronze Age, late Neolithic, as well as potentially at the end of the Bronze Age. We can also say that the late Neolithic and early Bronze Age strains were likely not bubonic, which is interesting because we need to understand how it was transmitted. We really have no clue right now. So by that, I would like to thank, of course, my whole department, the many collaboration partners that participated in this ancient pathogen research, and the funding agencies. And thank you for coming and sharing this happy event with us today. So thank you.
Thank you so much, Johannes. And I think we have about at least 5 minutes for a few questions.
Malcolm.
Johannes, thank you for that wonderful summary. It's perhaps worth pointing out that we have textual evidence for the plague in the last half of the 14th century BC.
Yes.
2 successive Hittite emperors
died.
Plague. there's one recorded text where the Hittites beg that the neighbors bring a statue of the sun goddess Arena to the Hittite capital of Pokeskoi because none of their gods have been successful in stemming the plague. And there's the first recorded incidents of deliberate warfare where the Hittites and the Assyrians each try to send infected rams behind the enemy lines and rams carrying bloody.
Yes, that's a very good point, Malcolm, of course, that there is historical evidence for the Hittite plague, that there was a plague outbreak in the 14th and 13th century there. And of course, you know, Yersinia pestis might now be a good candidate. Of course, in the past, people couldn't link this up because we didn't know that Yersinia pestis was even around during that time. But now we actually do know, even though first we thought the Stone Age plague is kind of not bubonic, it's kind of different. But now that we do have evidence from close by, I think it's a very high chance. The unfortunate circumstance, though, is that the Hittites didn't leave their bodies behind. So we have a big lack of dead Hittites. So if some of you archaeologists in the room stumble across some dead Hittites in the future, please let us know, because they're very, very difficult to find, I have to say.
An interesting question is what ended the pandemics particularly. So if I remember, you said that by sequence of the Black Death isolates are identical to the last nucleotide. So therefore, they cannot be attenuation of the pathogen. Are you suggesting that it's improved public health measures or evolution of the immune system that eventually limited? So if half of Europe die, why didn't the other half?
Yeah. So the question is, what basically stopped the pandemics, and why do you see basically that maybe the pandemics will pop up again? I think it's a mixture of both. It's for sure, it's cultural evolution. People started quarantine. People kind of had more, basically, kind of public health measures to try to eradicate maybe the hosts, so the vectors like rats. But it's also the pathogen in itself, because if you kind of conceive the disease, your immune system, your adaptive immune system, will actually give you lifelong immunity if you survive. So what happens is, if you have a massive outbreak like the Black Death, probably the entire population of Europe was infected. Half of the individuals survived because they were lucky enough that their immune system actually adapted, but then they were lifelong immune against it. So it's the same what happened with Ebola 2 years ago in West Africa, where quite a few, about half of the people that were infected actually survived. They are now lifelong immune against it. And they were actually the people that they now use to treat patients that then came in later on with the disease, because they knew those people are immune. And this is why it goes through cycles through time. So you basically have an outbreak, then most of the population is immune. Then, of course, they cannot inherit the immunity to the next generation because it's adaptive. So the next population is vulnerable again. It's naive again. There's a new outbreak, and so forth. So this is why it goes through the cycles. One question then, of course, is why does it disappear in the 18th century for good? And that could only be because basically those public health measures, quarantine, hygienics come in and then probably eradicate the vectors, and therefore then the basically disease disappears.
But there is possible to study that, because the immunoglobulin, or the markers of the adaptive immune system, can they be traced in ancient human DNA to confirm this hypothesis?
Yeah. I mean, it's very difficult, because you have to imagine that after those hundreds or thousands of years, there's very little DNA left. Now, DNA has the very beautiful property, you can amplify it. Actually, everything that we do involves a lot of amplification of the DNA. We start with tiny, tiny amounts of DNA, often less than a genome, so which is really like picograms, like tiny amounts that we can amplify to micrograms, like a lot of amounts, because sugar cubes of DNA, if you want, because of the polymerase chain reaction that allows you to amplify DNA for proteins that doesn't exist. So the proteins also degrade through time, and there's so little protein left, especially of the immunoglobulins that give you immunity against a certain pathogen, which are in very low concentration. They're there, yes, but in too low amounts that you would easily find them, because you have a hard time to even find, sometimes collagen or some of the most common proteins in human skeletons. So it is much more challenging to look at the kind of tiny little protein traces that you might find somewhere in the skeleton. Of course, theoretically, you should have maybe T cells or B cells, which would still have basically the kind of makeup of the immunoglobulins present. But again, they're in such a tiny fraction of the total DNA, and the DNA is hacked into small little pieces of about 40 base pairs on average. You couldn't even span this kind of variable region that you could reconstruct to then have basically some proof or evidence of this adaptation. But we talked a lot with people here and other people that would be wonderful to do something like that with proteins, maybe in the future with new technologies.
A final question. Just a small comment on this from the archaeological historical background. What is also cross-fertilizing with our archaeological background is that we can now see the spread of pathogens through different times. We can model the spread of this. Now we see that, for example, it's always the big centers that are hit at the Levant. It starts from the north, goes to the south, and it's always the same in Al-Arab, the same as in Hatzor. We see in the last decade of the city, we see a decrease. We see that things are given up, partly are destroyed, as in Hatzor, the gods are beheaded. We see crises, and then the big centers are given up. And all the small centers around, they just continue as if nothing had happened. And this is so interesting if you compare with other times, you know. What centers are hit first by this kind of spread? What is the background that you need for a disease to spread effectively on the population? Now we have a chance to, by crossing, this is what I think what makes MARM so unique, that we can really connect these different perspectives and study this kind of spread of diseases that one discipline on its own could not do. And this, I think, is, for me, the biggest profit from just, you know, so many questions arise from your presentation for me as an archaeologist and vice versa.
That's good. We have 5 years now to address those.
And now there will be an actual final question, please.
Okay, this is the final question. So the genomic data on the prehistoric screen is really interesting, and you focused on its ability to be hosted by the sea. And then you said it wasn't a Bubonic plague. So are there genetic deletions or genes missing that are responsible for variants? For example, talk about adaptive immunity, but the way plague is probably successful is by foiling a specific component of the DNA.
Mm-hmm.
So with the genes that are responsible for that or other virulence genes missing, it becomes important because I'm trying to figure out if human migrations are associated with the spread of plague the way you describe it, or whether they're responsible.
So it's a very good question. So are there other adaptations that we actually see? There is actually one that is quite interesting, actually kind of something that you're almost referring to, and that is that we see the flagellin genes are actually getting lost early on during the evolution of Yersinia pestis. So we actually, the closest relative, Yersinia pseudotuberculosis, actually can build a flagellum, which is something that the immune system, the kind of innate immune system, has learned to attack with the complement system extremely efficiently. So Yersinia pestis has to evade that, but he loses those genes. And we see that actually multiple lineages happening convergently through time, which is maybe something that you're referring to, but we can also talk about that kind of afterwards if you have more questions, okay?
And by the way, tell the people in Madagascar that the Bubonic plague is that important.
I mean, it is true, of course, there were cases in Madagascar, there were also cases in southern Asia, but I mean, compared to the bubonic cases that were reported in the 20th century, it's a minor percentage. It's probably more in the promille than it is even in the percent, so it is quite rare. Sorry? With a different death rate, of course. I mean, the death toll, of course, is much higher because we have antibiotic treatment, which is quite powerful with bubonic plague, but of course, with pneumonic plague, it's much more difficult.
Thank you so much.
No, I don't have. Okay. No slides, just me. Turn on the lights, open the windows. Oh, mic me up like I need it. All right.
Thank you. Where she is special counsel for institutional outreach and engagement, as well as professor of classical archaeology and classics. She will be speaking on impact and opportunity, forecasting futures for the work of MAM.
All right. There is, I'm sure you'll all agree, always an excitement to attending an event such as this. Not that they come around very often. There's a realization that you're watching the launch of something that could be special indeed. To a great extent, efforts in the academy tend to work on incremental scales. You tweak this, you shift that, you grind on, and there's nothing inherently wrong with such a method of proceeding. But every now and then, you really want to heave a very big rock into the middle of a very important pool and watch the spray fly and the ripples rip. And that is, I believe, the Max Planck Harvard Research Center for the Archaeoscience of the Ancient Mediterranean is on its way to becoming just such a rock, perhaps a boulder. Now, that may not sound like much of a compliment, but it is intended very much to be so. Now, whenever you get paid a compliment, of course, you frequently run the risk of follow-up, a follow-up request, a dace, spectacular, congratulations. But have you thought about adding X, doing Y? What about Z? The more the praise, the higher the expectations. So as a brand new member of the Center's Scientific Advisory Committee, I figured that my role in the conversation today could be to look down the road in light of such hopes and expectations, and to forecast some of the Center's possible future directions. My vantage point, I should underline, is from the archaeological and the North American side of this cosmopolitan and interdisciplinary house, so please forgive the biases. And my forecasting will take on two very closely related forms. First, on possible impacts, broader impacts, watching how the Center's work could reach out, ripple out in the widest possible circles. And second, on the opportunities granted by its rare mix of scientific and humanistic inquiry into significant and compelling subjects. Opportunities, perhaps, that even tilt into the realm of obligation. Now, as with all forecasts, especially those prognosticated in early days and from early readings, and these are early days, the ink is barely dry on the page, these forecasts may prove wildly inaccurate, and as with many predictions, they might come as a bit of a shock to the people involved. But I truly do not believe that that is the case here, not with this team, as I'm starting to learn them, and I very much hope not, for reasons that should be clear by the end of my pitch. All right, forecasting impact. The high-end research impact of the Center is already clear and present, beautifully showcased by the talks that we've just heard. I could sing praise of the virtual connections to be forged. We really need good models of how to shape such rich and efficient international coalitions. The inclusion of the next generation, the graduate student support, the young investigator dimension, that's all wonderful. More compliments, which means, of course, more, but what else can ever more expectations. And now, while I understand that MAM, when fully spelled out, is a research center, I also take note that just as the P of Planck is somewhat mysteriously missing from the acronym, so is the R of research. So I will take that as a sign that research here is seen as an enabling and not a limiting term. And therefore, I'd like to pursue how the Center's findings could reach audiences beyond the immediately scholarly and expert, could stimulate other conversations, could locate unexpected allies. Now, in the interest of full disclosure, this concern, if that's the word for it, stems in part from my current position, as you heard, in the Office of the President at the University of Michigan as a special counsel for outreach and engagement. But more fundamentally, I have always been grateful for the forceful convening power of archaeology and the study of the ancient world, our ability to attract broad spectrum and passionate communities of interest. Now, this center is far too savvy to underestimate its appeal, the astonishing appeal of the types of research being done and the new stories being told. Climate change, disease, falling empires, plague, mice, the Iceman's ulcers, plague, shipwrecks full of bodies, little old ladies running around acting as agents of technology transfer, plague, alpine marmots as bioweapons, Vikings in the Canary Islands. I mean, you can't make this stuff up. And this, this is incredibly sexy, provocative, material. And one, whether you will or not, it will make a powerful splash. That, I would argue, is something to be captured and employed. Now, numerous populations could be involved in this enterprise. Students, as the principal agents of change we send into an unsuspecting world, should always be first and foremost. Graduate students, clearly already in the center's DNA. Undergraduate education is notably on the radar, at least in the initiative for the science of the past, and I'm presuming that will infect out into the world of the center and its participants. Now, among the many forms of diversity we seek in undergraduate learning environments, breaking down that stubborn divide of the two cultures already referred to, arts versus science, we cannot forget this. We experimented in this space at Brown University when I was there, if more in the material than in the biological sciences, and found it cheerfully productive. Again, though, thinking about undergraduates, the peculiarly edge-of-the-seat quality of the center's work is extremely compelling, offering not only an attractive, but a sort of a culturally complex, juicy kind of on-ramp to a genuinely integrative education, an integrative STEM plus education. So in terms of impact, reaching college students, I think we're good to go. But I wonder if we can push the influence of the center harder and earlier than that. It's increasingly clear from an ever-growing body of evidence that when it comes to turning many children onto subjects such as biological science, let alone ancient history and archaeology, waiting till college is already waiting too late. This is especially true, of course, for kids from less privileged backgrounds. So we have somehow to stimulate and feed a pipeline of awareness. And here again, I think the nature and the appeal of the center's inquiries could be tapped. Tapped by creating digestible, not dumb, but digestible translations to pre-college learners of all ages, K through 12. Now, this is difficult, but not impossible, and it would involve finding ready allies in perhaps some surprising places. I envision you recruiting a graphic artist to tell the story. Mickey Mouse does the plague, something like that. Capture their attention. Use it, use it, use it. The traditional barrier that tends to exist between higher and lower education, at least in the United States, is today being very firmly challenged, not least with new technologies for sharing content and more enthusiasm for translating content, and that's across the entire educational course. And there is finally the wider public, the non-enrolled public, the dearly hoped for and much to be encouraged population of lifelong learners. I'm a personal believer in reaching out through massive open online courses, MOOCs, and using other evolving forms of online interaction to diverse and distributed audiences. Just as far as impact goes, I taught such a MOOC called Archaeology's Dirty Little Secrets, which in 2 iterations of 8 weeks each touched as many learners, some 30,000, as it would take me at least 300 years to hit in the classroom. And that's a conservative estimate. And that is just raw numbers in terms of age, life experience, nationalities, to a degree, educational backgrounds. One is there engaging with entirely different classrooms, populations, with a chance to loop them back and forth, not only with each other, but back to our students on campus, our residential populations. In other words, you're given the opportunity to play around with new and unbounded pedagogic paradigms. So, and going in this direction for the center is a natural straight shot, given Harvard's partnership with MIT and edX. Now you'll notice how generous I am being with the center's time and the center's money as I spread this wealth around across multiple and diverse audiences. And some still might say that these are constituencies that are not the business, not the problem of major research universities and high-end research centers. I would argue, to the contrary, that in the grand scheme of things, it doesn't take much investment to make available superb, already done research, and that bridging results out to others can offer its own unexpected insights and rewards. Not to mention forging and finding additional collaborations across the academy. Nor am I sure that any institution of higher education in this day and age can, let alone should, fail to see the wisdom of investments that work to promote public understanding and regain public trust in just what we do. Now, I said my second set, that's impact. I said my second set of forecasts would revolve around opportunities, verging into obligations. Let me start with an obvious statement. Many of the topics core to the center, climate, migration, genetic tracking and analysis, pandemics, disease vectors, all are current and urgent topics to our 21st century world. Let me follow that with another obvious statement. There is a profound crisis in scientific literacy in the United States and beyond. I do not know the situation in Germany. And let me add to that yet a third obvious statement, which is going back to the level of inherent interest, the sexiness of the center's work, and the bandwidth it is likely to draw. Put those three together, shake well, and one is left with an unmistakable, if daunting, opportunity. Opportunity because there's a crying need for the insertion of expertise and evidence-based thinking into public conversations around these kinds of subjects. Nature abhors a vacuum. And if the knowledgeable don't speak out, fake science, fake news will eagerly fill that void. So there's opportunity, desperate opportunity. But daunting, because there's no question but such topics lend themselves easily, readily, almost automatically to politicized thinking and ugly assumptions. No one likes to get trolled, and you will be if you raise your head above the parapet. Yet that's exactly what I envision the center's participants will do, and do with aplomb, with authority, but also perhaps with an unusual advantage. Like so many others, I've always believed that the study of the ancient Mediterranean world can provide a kind of a go-to space for framing and reflecting on modern conditions, a chance to consider long-term developments with a measure of detachment and some time depth, some hindsight. Looking through a distant mirror can cool and balance conversations both within the academy and beyond. And when it comes to introducing, as you guys are, introducing potentially sensitive topics, not to mention an unfamiliar toolkit of fast-evolving, unbelievably fast-evolving scientific techniques, then such a space can perhaps allow a little bit more room for education and a little bit less room for aggression. To put it another way, providing the opportunity to understand how we study certain issues in the past might provide guidance and understanding to their investigation and interpretation in the present. And sometimes it may even boil down to saying, we still don't know yet for sure. Let's give it time. Now, this rather admittedly wistful view manifestly does not always work, and nor really should it always work. Even when we're talking about ancient things, matters blow up. Just within the past few months, there was the Twitter storm over the possibility of an individual of African descent living in Roman Britain. It got nasty, threatened to sort of boil down to the science versus the ancient evidence, meaning the texts, versus political correctness, and to no one's particular edification. Closer to home, there have been the reactions around the Nature article about Minoan and Mycenaean genetic origins. And everyone has other stories, I know, of misunderstanding, ignorance in many cases, and accusation. I hold no brief here, but these kinds of confrontations over the nature of newly derived evidence and their complexities of interpretation, these are not going to go away anytime soon, and that's both inside the academy as well as very much outside. Now, one could, of course, simply batten down the hatches, plunge on regardless, don't let any of that raid on our parade, and that would once perhaps have been a likely academic outcome. But early days, though it is, it's already manifestly clear that MAM and its leaders have no intention of taking that easy way out. My impression is they are framing, starting early, but framing very much their work in a very different way with responsible responses to these very palpable needs and pressures. So for what it's worth, I predict, I foresee some positive landmarks on the horizon. I see a range of populations being exposed to and their education and worldviews impacted by this work. I see opportunities offered to be carried on, God bless them, by your students, to model responsible scientific communication in the public domain. And I see the creation of a new and effective platform for blended humanistic, scientific, and ethical conversations, not only deeply relevant to our understandings of the past, but also directly implicit in the contours of our future. Now, I realize I am heaping here a very large burden on a very young baby, but given the combined forces of Harvard and Max Planck, with all the smarts, bully pulpits, talents, and resources, I foresee a team who can make these crucial outcomes come to pass. Please take that as a compliment, a huge and heartfelt compliment, but we'll be watching with hope and expectation to see what you do next. Thank you, and good luck.
Thank you so much. I especially appreciate your comment on Mickey Mouse as a plague vector.
It's a natural, isn't it? You know, kids love gruesome. So questions? It's sir.
I'd like to make a comment.
Uh-oh.
It's about the difficulty of not having your work used for evil. So I'd like to report a conversation I had with Svante Pääbo some years ago. We had discovered archaic admixture into various human groups, and some human groups had more archaic than others. And Svante was rather concerned about the possible political reactions to this. And I commented to him that if you're a racist and you find out that your group has archaic admixture, then obviously archaic admixture is good. On the other hand, if you find out there's the other guys who have archaic admixture, then archaic admixture is bad. And I don't see what as a scientist I can do about that, really. I mean, and in fact, that is the kind of reaction that one has got from some quite unpleasant groups in our society, and there'll be more of that. And you can try and answer it and say that's not what the science says, but somehow you don't always have the megaphone.
I guess what I'm saying is we have to take the megaphone. I think we do have, well, those with expertise have to grab that megaphone and start trying, and in our teaching, if not in our public discourse, trying to shape people's understandings of these things that they'll read in very, and I'm not saying it's easy and it won't happen overnight, but I think just to stand back and say, we're just putting the science out there, let the chips fall where they may, it's one course to follow. I think it's an extremely, I think,
But that's not what I said I wanted to do.
I didn't think so. No, I think this is, and I'm sure this is a conversation that is going on all the time. How does one, given the nature of some of the problems that you're discussing in the past and, present day analogs or even recent analogs, I think we have to be careful about what we say, how we put it out there, and how we frame and contextualize it. Sir?
You seem like such a fun-loving and enthusiastic person that I thought I'd share some things that you can occupy your spare time with. There's an excellent online course on play out of Purdue that's in great courses. And it's,
Fabulous.
Brings in a lot of very important themes that are not
Super.
going to be emphasized necessarily in this group.
Great.
About art, play, saints, faith, and so forth. It's really, really good. And along with Mickey Mouse, actually, there's a very good archetype for that. The malaria project during World War II, as you probably know, recruited Theodore Geisel, Dr. Seuss, to use behavioral change ideas to persuade the soldiers to put on mosquito nets and long pants and so forth and so on. So there are precedents we might learn from. They're kind of fun.
Thank you, sir. I didn't know that about Dr. Seuss, but I am not surprised. And I will look into that. I floated around a little in the MOOC universe and didn't see anything immediately, but it is such a large. Well, then it's not a MOOC. You know. I'm sorry. Other questions? I know it's almost reception time. Sir.
One item of good news, we're not alone. On December the 8th, 2 months from now, right here in Cambridge, the American Academy of Arts and Sciences has convened a preliminary meeting on the whole question of past episodes of climate change and their relation to famine, migrations, pandemics,
wonderful.
And warfare. And assuming that goes well, it will become a sort of a major topic with good funding behind it and an issue of daedalus devoted to it.
Fabulous.
And universities are starting to pick this up. I would mention especially the University of Arizona, which has the country's leading, in my view, leading radiocarbon lab and leading tree ring lab. They had focused their activities on the New World, especially the trees, the bristlecone pines, and foxtail pines in Nevada and California, which lived 10,000 years old, make nice annual rings. But it came to think that the real problems really were planet-wide, and hired some classical archaeologists and created a new program called CMATE, which stands for Center for Mediterranean Archaeology and the Environment. So they can bring together the work of their outstanding department.
That's convergences. That's wonderful.
Lab, other aspects of the university. But the interesting wrinkle is that they're taking undergraduates as well as graduate students in the program, subject to the understanding that each one will have to specialize in one area of history or archaeology or anthropology and one area of physical science. And then they'll get joint degrees. And when I first heard about this, my first fear was, my God, where are these people going to get jobs? And now I look to the day when, without that sort of joint training, no one will be able to get jobs.
I think that's very percipient, and if we trained only to get our students jobs, we would have been out of business a long time ago. No, Mike and I had spoken briefly about when do you start this kind of training, when do you start this kind of intersection, and I think, you know, waiting till graduate school, unnecessary and unhelpful. I have to say, it's so interesting to hang out with a group like this where you can talk about war and pestilence, and just say it's so exciting. It's it's not usually the word I would use, but other questions. I think we're good.
Thanks so much. You get the final word.
Oh, I get the final word. Okay. The final word is. Thank you all for being here, for participating, for listening, for asking these great questions, for being such a wonderful, thoughtful audience, and having contributed to this grand moment of the birth of a new institution that is aiming to be an infrastructure for a new discipline, for being part of it by your presence here and being witnesses to that. And after all the hard brain cells you've devoted to the last several hours, I think you've earned a reception, and it's sitting here waiting for you. Thank you very much. Thank you all.